Now we're connected. Okay, live from Brazil, it's Tuesday night. Yay! Tonight's special guest stars are the plankton.
Heck yeah.
So we are at 304 meters on our way down. We are looking for interesting things to collect, both for morphological and genetic analysis, to try and understand the biodiversity in this stretch of the ocean, which has been very poorly studied beyond the thin surface layer. And you are going to see some of the most amazing midwater animals you've ever seen.
It's over here writing checks.
Oh, yeah. Yeah, send your checks to them. We will, in particular, be focusing on gelatinous zooplankton, which are understudied globally, and which are extremely difficult to assess with traditional tools such as plankton nets. Okay, do we want to slow down a little bit and start working our way down, or did Karen say she wanted to go to the bottom and work her way up?
I think it's just because the shorter dive, just.
Okay, all right, then let's go to the bottom and we'll work our way up.
What did she say you wanted, mostly?
Doliolas, larvaceans, and then anything else that we think is good. And she said to go to 600, right? Not,
600? Okay.
not all the way to 8.
Yeah.
Pessoal, boa noite. Estamos ao vivo na oceanographic Falkor.
All right, that's better. This way we can see something.
Eu sou o Marcelo Melo, professor da universidade
All right, so let's try and keep
de São Paulo.
a little bit down velocity as we go.
E a gente está aqui na sala também com o professor Ross Robertson da Universidade do Alasca.
That's squids.
Ah,
that's
peixe.
cyclotone.
I think it's a cyclotone, Mictofi.
Let's see what's out there. I think it's just a larvacean house. And I think it's empty.
What was that just now?
What is it? It's a larvacean
Chegamos
house.
à nossa primeira água viva
I
azul.
think it's a fritillaria house.
Aparentemente é alguma coisa que já está
Okay,
morta.
let's continue on. Red object over here, or is that that's an artifact? Oh, okay. Too late.
Shrimp.
Shrimp?
Shrimp?
Is it
Yep.
shrimp or a fake shrimp?
Shrimp.
Oh, camarão.
Long antennae like that, probably a suggested. When you get these
Suggested.
antennae that are held over to the left, when you get the antennae that are held kind of forward and then trail straight down on the sides, that's usually a suggested. I think we're coming back to it. So passing it around. Here we go. Looks like, oh, it's a big one. Wait for it to swing back. We can see it in the upper camera. Who's it?
Yeah.
Okay. That was some type of large hella created, I think. That's a Larvasian house, I think.
It's actually, can we do
Yep.
this?
Okay. Which do you find easier? Is it easier for you to stop coming up or stop going down?
Now, it depends on some a little bit the training capability is different. So yeah. But, normally, say going down is better.
Okay.
Estamos agora no nordeste do Brasil, mais ou menos em frente a Alagoas, ao largo do Nordeste, na verdade, a cerca de 350 quilômetros da costa.
House. So the difficult thing is we can tell the house looks like it's probably recently used. We can't be certain most of the time. It's an animal here until we zoom in. They're abundant enough we can't afford to do that with every house. There we go. Up on that, it is a ctenophore. It is this mertensia that we've been seeing several times and keep losing.
Say that one more time?
It's a ctenophore sediped. It's in the family Mertensia. One of the things about mertensias, many of them are kind of flattened. So unlike true jellyfish, the siphonophores simply have sticky cells that they use to sorry, the ctenophores, sorry. Ctenophores just have sticky cells. They actually don't have harpoon darts inside their stinging cells like a true jellyfish does. These were once put in the same phyla, and about 40, 50 years ago, they separated them into separate phyla.
What is the family name?
They separated the cnidarians out from the
CY?
ctenophores. They were both in a phyla called Coelenterata.
Huh? PP?
Nice.
Thank you.
So what we want to see, okay, it knows we're here. So this will provide a nice chance for us to look through the animal now that it's moving. It really does seem to want to keep that one axis flat relative to gravity.
A tiny shrimp following us.
The back's their Medusa, right?
Hmm?
The back there is that a Medusa?
Yeah, there was a, we went through
Esse é
Medusa, we
ctenophoro.
went through another siphonophore a little while ago too.
Ctenóforos são organismos planctônicos. Dá para perceber, dá para reconhecer por causa desses feixes de cílios que refletem a luz e dão essa
These
aparência.
have actually been holding up surprisingly
falta
well
cor ali,
with
né?
the suction sampler. So we don't have any of the scanning we need to worry about. All right, that's some nice video. Let's go right to a sampler.
Yeah.
Yep, suction sampler, please.
Para quem está perguntando, isso não é uma
I think we
medusa,
want to save
é
the
um
D samplers
ctenóforo,
for
é um
things
outro
that are a
grupo
touch more
de
fragile.
invertebrados.
So the same thing, once you think it's partway down the tube, you're best to cut the motor off.
E a gente
and if
consegue
we need to,
reconhecer
we
eles
can flick
facilmente
it
por causa
on.
dessas feixes de
little
cílios que
Yeah, first I need
fazem a
to-
locomoção.
So that air to get out of there, yeah.
There. Close to the too far out. Is that okay?
Are we collecting this one? Ah. Então a gente vai tentar coletar. Isso aí vai ser a nossa, se der certo, deve ser a nossa primeira captura da noite. O ROV SuBastian é equipado com esses equipamentos especiais para coletar esses organismos. Esse é um funil com a bomba de sucção. E uma vez coletados, esses organismos são armazenados em garrafas. São chamadas de canisters.
I see it coming here. Yeah.
There it is.
It's in.
Suction off.
Então ele entrou no canister, agora o canister fica fechado e vai ser trocado por outra garrafa. Essa é a garrafa de número 1.
Index number 2.
What was that?
That was one.
One. Donkey first.
UTC. Oh, UTC's hard to talk.
That's the quickest collection we've ever done. Okay, out there, up. Dead center. Let's have a look at that bison neck.
Mais
think it's
alguma
a
coisa ali na
neck.
frente. Ah, peixinho passando.
Or is it a calicoffer? No, it's another cyclone, another little, teeth.
So do we want to?
We saw this with the blue, with the greeny area on it before, right? Needed to get a little crisper for me to see exactly what we got. Yeah.
I mean, I took a look at it. I have access to. So I'll modify that. But it's really a shame that we can't move that.
It's not swimming like.
So the question is, do we want to just slide sideways?
Okay, get a frame grab of that.
Yeah, and then we can see ourselves somehow.
Look,
Yeah, no, I'm just saying, like right now.
that.
What is that?
Did we collect this before, or we just saw it?
I feel like we tried and lost the place we will have one.
Okay, let's collect it then.
Collection.
What is that, Ross?
It's some type of isophenophore. I can't even tell you for sure. I'm not seeing the end of it to tell you if it's a bisoneck or a calicofferum.
So, but that would only do one.
It looks more like it's a calicofferum.
My thinking is we start with just one and then find out how down the lot. Center there, it's coming up. Either way, we're just.
Okay, did we just fragment it?
Então esse é um sifonóforo. Sifonóforos são medusas coloniais. Cada célula, né, cada indivíduo é adaptado para uma função diferente. E a gente vê um tipo de enguia ali na frente também. Provavelmente Cerrivomer.
Alright, it's right there. It's right up its it.
Got 60. Set for a certain size. It's designed for setting for a certain size.
E essa vai ser a segunda captura da noite.
And then switch
Did we get it?
size and moving the wheel back to its block, basically, so that you have a few distinct ones.
Easy to see rust also on this.
Oh, you got it up on this screen. Okay. I keep moving around.
The limitation there will always be you can only have these.
Oh, it's not showing up.
But that's where the philosophy of to take or remove the two different approaches.
Looks like we got a phroneme in a house maybe in front of us.
Tank holders.
Some type of an amphipod.
Right.
I have not seen anything go in. It takes, what, about 30 seconds?
Sure, she is able to dishes
Oh,
and
it's
stuff
after
too.
a bubble shed. Yeah, I mean, there was a good argument there, the same thing, you know, that people also like to pose the
something's going on there.
which you would not have that.
Maybe it's the soul.
So yeah, that might be it.
That is definitely true, right?
It was pretty transparent, so I'm not sure. Something seems to be in the bucket, but I don't know if it's just air.
No, but you were just saying.
If you need different magnifications, you move your cameras in one go to the next.
How confident do you think you had something down the bucket?
Fifty percent sure.
You just twist it and see. Check.
Larger field of view in between.
I haven't seen any.
Resolution kind of makes
We
up
would
for
have seen it go in, at least.
I saw bubbles.
I just saw bubbles is all I saw go in.
I guess I'm not really concerned about the operation. As long as you're, you know, moving or making. That's like exactly how I want myself too. Just get it off to the right.
I would do the same thing, the same way every time. I thought it was totally weird when it was put because
Let's rotate anyways,
but I
maybe.
think with some effort in that there's like even a calibration cross. to
I'll
make
take
sure
the,
that it's actually
I'm tempted to leave it.
right on camera.
Leave it
It's
where it is and we'll
focused.
stick something else in there next just to be
It's not
sure.
really for the cameras because it's
I'm not
gonna be
confident. I didn't
higher
really feel I had,
percentage if it was in one
I
camera.
thought anything go in.
But you're also not going to be turning the focus ring on 3 cameras simultaneously. So,
Not unless there's something smarter going on.
yeah. So, for the top and bottom cameras,
Okay, over here,
having focused a little bit.
siphonophore.
Tirando a nossa descida por aqui, estamos chegando a quase 390 metros de profundidade.
Right there.
Move the camera back and forth, which maintains the magnification.
Acabou de passar uma lula, rapidamente deixou o rastro de tinta que a gente consegue ver
It's
na
the same
frente.
thing. It is the same thing.
Sandwich your
Ah,
Yeah, it's got the green.
It's got the green thing. Okay, so let's try again.
Extended.
All right, not too much.
achou o mesmo
Right up front,
organismo.
too. Alright, I'll come in a little more. Coming in. Do like right there.
Okay.
The
some
siphonophores are colonial jellyfish. They're connected together
that's
by
ink.
a stem. A part of the stem buds off new
Squink.
individuals, and the colony gets larger as it gets older.
Angle bed.
I saw another one.
Huh?
I thought I saw one that went off the screen.
No, that's a squid passed through.
There it is, right there.
It got scared and
So
released the ink.
We're just gonna rely on the
It's easy to do.
Did you get in?
I think it went down the back side.
Not sideways.
Upward,
Right,
no.
for sideways, right. That's. I was thinking of slotting it so the tank can move, slotting it so it's 2 rolls. Good is probably a better idea. Do
Going
that
upward, upward. Right, and then have a- where it goes, can you just be kind of-
Is that it right there?
Here it is.
Yeah,
That one?
Right there.
Either that or the one above it. That's actually it up here,
That one,
isn't
yeah,
it?
Yeah, it's all right.
there. Which twin side did we go to bigger one?
That's it. No, it's-
I don't know if it's gonna be a metal watch.
I think it's this one. I think I see the green.
This.
That's the one, yeah.
I mean, I can't put the
It's tall, tangled.
run it.
Well, what are you trying to do? It's like it's very difficult to get into and then take this dog.
The other thing is like the sharp, because of the sharp corner, it's flat down. Unless this is up to this.
Hmm.
So you've got two
It's better than
lines.
the foreshow, but that's what you're able to like sort of mask the light.
Just above. Yeah. I was just trying to put the light around to see if we can honestly.
They were refused with the other light, which was fine. They were refused with the other light. This was fine too.
Also,
Right boxes.
I guess too close and
Yeah, it's almost too. The glare is just in an awkward spot on that. Just say it's still.
It's the same one.
And that is a palette. Correct, these are helicratic created.
Lights it up and strobe.
Nope, but maybe we'll run into
Right.
them.
An additional white boxes, but not much of a very difference.
We want to get that. If we feel we've lost that, we can go and look at that jelly that just swam by. It is to the left a little bit.
There's spiders that I can't
Ooh, Fish.
What are you?
Oh,
A
cut
long,
of
thin
fish.
fish
Can I get a look at
brought
that?
in by the lights.
what we're looking for now. So.
Now we're going to. There was a big jellyfish that went over this way. We may have lost it because of too much of a delay.
I think it's like a trichurid. But I can see very well. It's gone.
You smell that?
Halicreated.
Spiders and fish.
Yeah,
Oh,
I got the
okay.
Then the best is out the move. So.
Yeah.
They're way close.
Bridge mission control.
Go ahead.
Could I have you guys move a bit north, northwest, please?
Yeah, already going there.
Thank you.
Thank you.
something in front.
Here.
They're not in front. That was. Oh, there it is again. The fish.
That's another fish out in front right now.
Oh, is that the guy again?
Looks like a swordfish or something like that.
No, that's something different. I don't see any green. This is a small prayed siphonophore. You can see you've kind of got two bells, one on each side. And it is a helicopter. I believe. I have been wrong before, though. I'll have to look. Now I'm starting to think it's the Piscenac. I'll need my phone.
We've got confirmation that it's helicopter from. Ooh, look at
That is one of our ctenophores again. Go after the ctenophore. Leave the little siphonophore alone.
that. Low bait Singapore.
It is going to be the Lassachilus or the Bathysauroi.
Yeah, what do you want for the date?
There was a small helicopter that went above us. A small.
Agora a gente tem aí mais um ctenóforo.
Okay, so
Né? Agora dá para ver os detalhes com bastante riqueza. Um bicho muito bonito, apesar de ser transparente, né? A gente consegue agora ver com muitos detalhes dentro dele. Todo feixe de cílios. Esses cílios não são bioluminescentes, eles estão assim só por causa da luz. Eles refletem a luz e lembram muito a purpurina que a gente usa no carnaval. Acabou de passar um copépode ali na frente também.
okay. That was the last kill us. If I didn't say so. All right, move along.
Ó, chegou agora a 390 metros. A gente consegue ver que à medida que o ROV vai indo mais profundo, a fauna vai mudando bastante. Tanto em termos de biomassa, quanto na diversidade também.
All right, up to that. There's mostly faunapore there, and I think there's a jelly up right back here, or a house.
When you get older, you will understand.
I already understand.
You already do. So far, I haven't activated my.
That's a
weird
house.
back.
Go for the target
Yeah,
behind
if I
it.
don't do this, I'll like turn my head and all of a sudden everything will just.
I've lost that. Might have been glare.
Right, continue to watch.
Thank you, Jimbo.
welcome.
Sorry.
House again. I think it's the same
I
house. I'm
know.
just going back the other direction. House. Another house. There. I think it's another house. Just swing forward and up. Right there. It's a see if somebody's
Consegue
home.
ver agora
We need
diversos.
to collect one of these during the dive. That one looks nice and intact. Okay, come in on it slow.
Agora tem um outro quinidário aqui na frente da gente. Outro quitenóforo.
I am not seeing kind of a white or opaque end to where the animal's trunk is. And I'm not seeing any refraction off of the tail. So I think that's empty.
Ou é alguma coisa de
That was,
muco
yeah,
também
that was
abandonada.
the Coelodendra. Or Coelo, whatever. I always get the wrong part at the end of it. What is that? That's another one. Coelo. And yep. Keep going. Out here, dead center, going up. Other
Center.
No, it's going to end up being, I think, Bathysceroi ortholacicillus again. I think it's just pulled up into kind of like the lips are pulled closed. Although we did get an Ochreopsis at one point.
That's a foot we should raise down. But also maybe.
Need to wait for it to open up. So I'm not seeing those rows with the diverticula. I'm wondering if it has a set of oracles that we haven't seen yet. That might just be this because it's younger. Okay, that's the last Kalis, I believe.
That's my understanding.
Okay. That makes
Yeah.
more sense.
Don't think it's larger than Medusa. It's just a house that's picked up a current and is swirling.
O
Continue.
Ross está tentando identificar.
It's mysteries. We wanna try and get as far the lab
Oop, there we go. Stop on that and figure out which one it is.
Do that for-
That's the same as before. It's a string without. It's like a lost
Some of
tentacle.
this can be exported directly from our.
Isso parece um tentáculo de algum organismo perdido. Não
Okay,
é,
continue on. I don't think it's living.
não é nenhum organismo
it's a
vivo, mas uma
tentacle
parte de
or
algum
something.
organismo que se perdeu e que está afundando.
I don't know how well that works if we're talking about still images, which you're taking from the match, right? I don't think you need to do this.
There is a solmissus to the right and another siphonophore, Physonex siphonophore.
Mais um sifonóforo agora. Legal que dá para ver os indivíduos responsáveis pelo bombeamento d'água funcionando individualmente. Parece um pistão de carro. E essa parte mais à esquerda é a parte reprodutiva. São os
A nice shape.
E os tentáculos são os de defesa.
It's not an apolemia.
Maris.
Your white Mars?
That's the undescribed species we caught a couple of days ago that I've been quoting Pseudogaiensis.
So you need or don't need?
We've got one.
Essa é uma espécie
You
nova.
could do a scan if you've got a.
Yeah, we can just scan.
You can do a scan.
Ela já
Do a
foi coletada em outros
scan, I can't
mergulhos.
Let's do one just in case.
remember. But some nice video and a scan, I think is
Well,
fine.
and I think we should.
E
Okay.
agora a
There's
gente
no
vai fazer
shadow
escaneamento
cam,
dela.
So getting Zillwards right now.
Right, can we start with Iris?
Iris.
Yeah, the one on the far left. We
Yeah.
just need to get it oriented
Yeah.
and play the lights.
Yeah, that, the one that.
So I don't think we want to do a
Where did he go?
We just disappeared. Does he? Should we do a collection or no?
Let's do a collection.
We already had one.
We have one.
Oh,
It's always best to have two of something because,
Okay,
as Dougal said, you can get abnormalities in a single specimen. And it's quick to process if we've already taken all sorts of pictures and we know what it is.
Are we really happy?
Não,
Então essa é uma possível espécie nova. A gente vai fazer a coleta, vai fazer o escaneamento dele e vai fazer a coleta.
Camera, the one is on the Irish.
Yep.
We have a feed.
Iris.
Vai ser escaneado agora pelo Iris, que é um sistema de escaneamento com múltiplos feixes de laser.
Are we just doing the one? Take a
Iris,
scan.
okay.
No, we're gonna start with this one and then do
Okay.
the laser. Like just kind of go through the process. Oh, what?
Yeah.
Hysteresis needs to be 0.
Just stop it.
Hey, my glasses. Oh, I was wondering
yes.
where they went.
I think the picture, is supposed to see on this one?
So,
It's
Google.
like down right there. You go.
your flat hortensia.
Where?
There it is.
You found it?
Right
You got one?
We think we got it.
there.
Cool.
Can you
Right?
get it a little
Can
bit
canister one?
further away from us?
All the way.
Yeah.
I hate it when this happens.
Not very much.
Are you a professional at removing Ethernet cables from connectors? There it is. This is it.
Can you do that again? I'm gonna adjust the color.
Alright.
Color map.
We'll be back in a second. We'll get a drink. This is going to be it.
You think I was blowing it out earlier? So it's a little bit left. What's this fish?
That's good.
Is it blues and greens or? Yeah, I'm only getting red, so if you can get it further away,
Far away.
there, it's getting better. Is the head pointed down or? I think it's there, okay. Wait, a little bit far,
How far?
yeah. It's got down a little bit.
Still in our view, it's just too far.
Got it, the camera.
Not the one where it was super far.
Yeah, still pretty far out. So the delta. Nope, just to the left.
É nesse trabalho de escaneamento para ter ideia
Should
do,
be
do,
fine, Russ.
de como os pilotos do ROV são precisos.
Oh,
Estão flutuando na coluna d'água a mais ou menos 420 metros de profundidade.
nice. A little bit closer
Eles
to
são
us.
responsáveis por encaixar um indivíduo ali entre aquele feixe de luminoso, né?
A little bit closer
Para que ele
to
possa ser
us.
escaneado.
Yep, a little bit closer. A little bit lower.
If you do the take that automatic box,
that's really good.
That's whatever getting.
Yeah, that's really good. Let me see if there's anything else.
Supposed to be blue.
Yeah, the focus is more on the head, which
like
is
the
low.
greens and
Low.
one's too close, blue is just.
Aproveitando para responder as perguntas do chat,
Well, I think we do.
a bordo tem pescadores de duas instituições diferentes. Eu sou o Marcelo Melo, professor no Instituto Oceanográfico da USP. E também lá do IO-USP tem o Marcos Reis, que é aluno de doutorado. E a gente tem a bordo também três pescadores da FURG, que é a Universidade Federal do Rio Grande, também do Instituto de Oceanografia de lá. São os professores Renato Nagata e Silvina Bota, e a pós-doc Julita Coerra, que é que mais interage com vocês em espanhol.
A little bit lower. You're so close. And to the left.
Desculpa, Julita, ser borrada.
To the left a little bit. Maybe a little high now. I don't know.
Ooh,
Oh, to the left.
nice larvaceans.
Oh, no, too close. A little bit more left. Maybe, I don't know. Oh, it's definitely getting it. But I think it's a little.
Esse é um sifonóforo. Isso é uma espécie de hidromedusa colonial. Eles são parentes das caravelas portuguesas.
I think
Então
it's
isso, na
too
verdade, é uma
far.
colônia de vários indivíduos. E cada indivíduo tem uma função diferente, especializada. Na verdade, tem grupos de indivíduos com funções especializadas. Então tem grupos que são responsáveis pela locomoção, tem grupos que são responsáveis pela reprodução e tem grupos que são responsáveis pela defesa. Então, apesar de parecer um indivíduo só, isso é uma colônia de vários indivíduos, chamados de sifonóforos.
Oh.
So it wasn't tracking right. Maybe it's not kind of part of this.
No, I think it's picking it up. It's just not displaying it properly. See? It's not part of
Well, we did record that, so
Yeah.
I feel like we.
Perfect. Let's do, can we do DPID now?
Yeah, great.
Yeah.
Great.
I was right. The Priidae are calicophers. It was word for a second. That's what we used to Google there. I don't need to know it.
I think Connie also said that it was a Caligofera, so I tried to
Yeah.
read it.
They're looking away from us.
Let it, like, go right through on its
It's
own.
in. Yeah, I know, right? Just
Amazing.
let it do it. Yeah,
I'm a guy. I don't
ideally,
believe it till I check it for myself. You
we'll
know,
want
where-
him to either be having his head pointed to us or sideways like this. So, yeah. Oof. Do you have copy pods for me?
doing the net tow until 2 in the morning.
But someone says that some copepods came in the
Oh, I didn't see
Oh.
them.
they are hiding for you.
They're hiding.
É, agora ele está sendo escaneado. Esse é um feixe de laser.
Oh, he's just to the right and below.
E ele tem que passar exatamente entre o feixe de laser e a câmera ali. Então ele tem que estar bem próximo ao feixe.
I mean, I could use that. Oh. Careful. Sure.
Tem que fazer mais um escaneamento de vida da colônia, na verdade.
Take it.
Frame?
I'll take it.
Right, so like
Yep.
all right.
E agora a gente vai coletar esse indivíduo.
We are gonna make this animal names.
A gente tem imagens muito boas, tem escaneamento, ele vai ser coletado e ainda a bordo tem um laboratório de biologia molecular, então vai ser feito todo o sequenciamento dele. E os pesquisadores aqui que trabalham com o grupo suspeitam com a espécie nova, então se for realmente uma espécie nova, a descrição já sai muito fácil. Com isso a gente vai juntando dados de localidade, de profundidade, da morfologia e da genética para poder conhecer melhor as espécies do Atlântico Sul Ocidental. Agora vem a parte que ele é sugado por aquele funil.
Do you want me to turn my laser on?
Esse funil tem uma bomba de sucção muito suave, né, para não destruir o animal, e leva para um container, para uma garrafa que fica acoplada aqui do lado do submersível. E quando eles chegam a bordo, eles estão vivos ainda. É possível ainda fazer alguns experimentos de bioluminescência, por exemplo, quando o organismo é bioluminescente, fotografar em alta resolução.
That's the thing. I really wish I had time to put all of the tests so effectively before. Yeah. But I had to do that here, so.
I was gonna say.
Oh.
Hi, Jerome.
This is true.
Huh. Sorry, I thought you were looking there.
Huh?
I thought you were looking there.
Oh no.
Yeah.
Oh, just low.
It's great. Yeah. It's gonna be fun.
It's big.
So that, yeah, sorry, the video data is gonna be a bit of.
Looks like there's a really big copepod, Dave.
Ai, entrou.
Slow it down. Slow it
Deixa eu ver se ele vai chegar no copo aqui.
Hi, Chris.
O indivíduo está coletado, está dentro do copo. Agora esse copo é fechado e ele vai ficar aí até o ROV subir de volta para o navio.
Bisonect. The genus we think is Marrus, M-A-R-R-U-S. But according to the experts, it is not described. So we will do that. That is the second individual of this species we've collected now for this expedition, which helps with the description. Now it's your turn to translate. Yeah.
Então esse indivíduo foi coletado, de acordo com os especialistas, é uma possível espécie nova. E esse é o segundo indivíduo coletado durante essa expedição, o que dá um bom número de indivíduos para fazer a descrição da espécie. Idealmente, quanto mais indivíduos a gente conseguir coletar, melhor para a descrição. Melhor para fazer a caracterização da espécie. A gente chama de descrever uma espécie quando a espécie recebe um nome científico. E a partir de então ela é reconhecida pela comunidade científica como uma espécie descrita. Esse é um processo extremamente importante, que assim a gente consegue compreender melhor, descrever a diversidade e eventualmente conservar espécies ameaçadas de extinção.
Yeah, like a clip. No, yeah, okay, there's the tail beating right here.
Isso é um urocordado, uma taliácea. Para perceber que tem essa parte externa e ali dentro tem um indivíduo. Ele secreta essa túnica transparente. E às vezes ele abandona a túnica, então a gente muito frequentemente encontra só a túnica flutuando, mas esse tá com o dono da casa, tá em casa.
And after we do that, I would like to stick it in the juice sample. It's a relatively newer house. It's not as quite as opaque as some of the others. There it is.
There we go. Very.
But if we hit it with a wake like that, that may be enough to have the gel. Just have to see if it's still there when it gets like that. Still there, I think.
Oh,
Probably.
apareceu alguma outra coisa ali agora.
Yeah. Oh, well, we don't want it. We're being attacked. They're pests. We need to. Thalassogalas, yeah. See the frilly stuff down the side? That is a big one. What's the size of it? Well, okay,
think
it's
it's
pretty
really
tight to the
close.
camera. That does make it look bigger, but it's still
Very close.
like the D sample mode.
It's very close.
No, you can even see him on the side scale.
I don't think that would be a good sample.
So 15 centimeters. No, there's siphonophore down here. Or is that a glare? Or something on the lens?
Tem mais alguma coisa ali que surge parecendo um fantasma na escuridão.
Oh, it could be.
Pode
There's
ser.
a nice house with it. Out on. Yes, that is the one that we just encountered. That's the proper one.
O sifonóforo que a gente encontrou agora há pouco. A gente acabou indo pra longe do ROV e agora tá de volta pra próximo da gente.
Oh, you're good. It was fun. We just used 1 and 2.
E agora a gente está na hora aqui do turno, troca de turno dos pilotos do ROV. Something in front.
Small jelly bit ahead or just the light hitting something?
Tem algo pequenininho ali vindo ali. É um peixe, é um hatchfish. É um peixe machadinho de prata, Sternopictide.
Okay.
Thanks, sir.
Run away. Right out here. And another. That's got awfully white arms. It almost looks like Colopanina. It's reflecting the light like one. Does this seem familiar to you, Kakana? Does this look like Colopanina?
I missed the photo. Are there white balls-ish
Yeah,
at the
whites
tips?
on all the tentacles.
I think there was something else that we saw earlier.
No, you're not looking at me.
I know. I
Like
wish
on
it
this
was
side.
It's coming around.
It's coming
to the
around,
front. There
yeah.
we go.
I know I'm gonna say something and then Dougal's gonna come in.
We haven't collected one of these yet.
Is it in rough shape after being
Well, let's see
kinda
how it looks
tossed?
once we get it back in focus. It hasn't dropped. Hasn't dropped tentacles. All the tentacles yet.
Yeah, I'm used to
Mais uma
Colopanina having
medusa.
a lot more tentacles, but I think that's a good guess. Are we kind of waiting for Dougal to rush in?
What's your guess?
Colopanina, which is what Russ came up with, but
Thank you.
you're welcome. I'm not very certain. I would probably leave it.
Oh, it's not next to it.
Can we get a 2-for-1? That looks like a little. Can't tell it's a little halicrata or a little
Yeah.
Arctic.
You know, like shaky.
How it, like how the Reyes. Did you want this?
Is this
Sorry, I'm troubleshooting. Is that a Colopanina? Looks like a Colopanina. Well, it's an animal that we don't have from this region for
Then let's-
genetics.
yeah,
so
let's
I
collect
guess
it.
suck it up. Yeah. We're not likely to catch it in a net anymore.
No.
So yeah.
All right. Let's suck them up.
That's a pretty broad.
George,
Yeah.
You haven't sucked up that one.
Okay.
Hey, buddy.
For sure, Rama.
I think it's too small for scanning, so I just.
Another Livestream house. And I don't know what the jelly is we keep seeing back behind it.
Is there a reason why we don't have the UTC tide on the side cam or the sea cam?
We should. It's just not on display or not looking the right place. It's not on the display.
Yeah.
It's added as an overlay after the fact.
Trying to suck up that jelly.
It's in there.
You know, it's probably just the feed you're getting. I suspect it is time stamping somewhere.
Well, it's happening somewhere. Just have to figure out where to look.
Oh, the red.
Just
That's the easiest thing to see.
Do that for you.
Otherwise, let's see, is there any
They
place
want
else?
the overlay on the side camera.
A gente vai coletar esse vídeo também.
Which monitor?
É,
Which place?
como que os pessoal daqui de borda não tem nenhum indivíduo desse do Brasil, dessa região do Atlântico.
Yeah, the computers are all probably
Ele vai ser coletado para que seja feita uma análise genética comparativa
They're local.
com amostras de outras localidades. O ROV SuBastian está numa profundidade agora de aproximadamente 335 metros.
435 meters, 8 degrees Celsius.
Look how low that oxygen
34%.
is. Yeah, that's low.
That is one of the lower ones. No, we've gone through the. We're coming up.
Jellyfish you're sucking about.
They're not high oxygen creatures.
No, but that explains why you don't see a lot of other stuff down there. Yeah, 34% is low.
Okay.
How dangerous do you think it is to plug the mass back in? Or find it?
We'll find another one.
I mean, we're fine for now, but we can do it after. If we're concerned. If you need to access it. Or something critical.
Nothing critical, but it helps me go through the data, calculate it.
We put it on.
Another sparkly radiolarian and another. That was a calicopteran siphonophore, Vivacian house.
Just wait.
It's not a lot. No,
Not
it's
sure. That was a jelly of some type.
Right after the dive, so there's something to think about. High revenue dives.
I can't believe we just had to restart it not once but twice. It doesn't seem.
That's the thing, I told you, it seems so obvious now.
Really? Why twice?
Yes. And one time helped a little bit, 2 times helped.
A little bit more forward velocity, so I've got time to see things coming.
Okay, you're not being serious.
It does work sometimes. It's incremental.
It's so terrible. Everything about it is terrible.
The outcome proves me potentially right,
I'm just relieved we didn't touch anything
but
on the
downside, yeah,
vehicle.
definitely pull out the interface can, open it up.
I did not want to touch any cables. God.
Well, I think you should have booted it 3
3 times. That would have been amazing.
Or die.
Like my eyesight is good, but like
It's a
with
sparkly
glasses, it must be
siphonophore
amazing.
again, or not siphonophore, radiolarian.
Move that thing again.
That was just, we think there's some type of radiolarian, but I don't think we have radiolarian in there anymore, so, okay.
Right.
Should check with Manu what we've actually finally decided they are.
I don't pretend to know. I don't pretend to.
Another house.
If you want my computer to like
Continua descendo agora. Chegamos a 470 metros de profundidade. A gente deve chegar até 1.200 metros de profundidade essa noite.
there's just 2 B's.
House, or sorry, up, siphonophore.
Mais um sifonóforo à frente.
I trust Wikipedia more than I trust AI
Which
overviews.
one is this? No, that's a, let's have a look at this. This could be a ctenophore.
E um ketognata ali na frente
No,
também.
not, this will be a Dougal. We chased this one last time and I can't remember what we called it. It's a Kelly Coffin, at least looking at that far. He was trying to decide if they were spheronectes, but I think he talked ourselves out of that. It's too arrow-shaped. What is straight ahead in the background? Let's just drive through that, through the background. There's a fish or something just behind it. Up just a bit. Right there, what's that? There you go. Up a little bit more and to the left a bit more. Was that? Okay. Let's continue forward slowly. Look at this. On. We won't get that back. Okay.
É o pessoal
Down,
dando
forward, slow.
boa noite do
Over
Uruguai, do Brasil, da Argentina. Muita gente acompanhando a gente da América do Sul. Boa noite a todos, boa noite a todas.
Arctic Demo would be my guess. No, it's Alacratic. I don't know which. So when Dougal goes back through all this tape, he's going to be complaining about my eyes.
And mine. Absolutely.
Yep, we can move on. Now I'm talking myself back into Archipelago.
Get over there.
Okay, let's mosey slowly. I'm looking for a larvacean house to collect.
Love it.
Well, nice, thanks. It's so good that you gave us few things. You can't see the one.
You can't see the what?
See the people.
Because the lens
Is too depth of focus. is
It's
too
like
big.
three
So,
cyclonicals. Small place next to it.
okay.
So one of all those lenses take you to an age.
It works perfect. Yeah. Often it's the easy answer is the best.
I missed it. I was looking at one of the other monitors. Sparkly protist.
I
Sure.
would say
Maybe we've been talking them. So check that one out. Ooh.
Oh,
This is a really large prize area.
Russ, can you enlighten us what the difference between the two?
It's
They rearranged all the phyla in the protists.
Yeah, it's a real can of worms.
And it's a can of worms because no one knows
I
what's
heard a
called
large
in
rhizaria.
Okay.
Did
So
you
So
guys
what
go?
is-
they're all
Nope,
under
it's
Okay.
spinning
rhizaria
around.
based on
There it is.
genetics.
Up in the upper
It used
right.
to be
and we'll be coming
forams
back
and
there.
radiolarians and actinarians, Phaeodarians, within that Phaeodarians and Colidarians and a couple of others. And now
So what
nobody
is-
really knows for sure. Most people don't know for sure.
They
how
just
fit together.
changed, you know, I think
Yeah.
they transitioned
Great work.
to the Cryozoa. Where did it go? Did we
I don't know. I
lose it?
turned my back for a second.
Yeah.
So
Is it
the
gone?
correct
Yeah.
term is rhizaria?
Rhizaria is now
Rhizaria
the
is the
group.
largest.
Now the
It's an
subgroup.
overarching group because we
Don't
are
know.
uncertain how things are related anymore.
But radiozoa and radiolarian, Wikipedia says the same thing, and that's my question.
no.
Radiolarian is the correct term subgroup,
Okay.
but now they split something into what is the Cryozoa, which now hosts many of these other subgroups. So they have elevated. Oh, that's the
Still-
Colodendrian.
I got radiozoa.
Is it pretty clear enough to you?
Radiozoa on
Yeah,
here.
this is
Yeah.
No,
is-
not perfect.
to be honest,
I was laughing because I didn't see your face. I
That's the
figured you were
same.
egging them on by asking.
What is that one?
No, but
No,
Manu-
I really
Yeah.
don't.
What is that
so
one?
I just,
Yeah.
I'm gonna
that's
say
Colodendria.
Yeah, it's super
Yeah.
confusing and they've made
What-
it worse.
What did we decide that
Colodendria.
sparkly one
Yeah, the
belongs
sparkly,
to?
I have an ID now. It's Hastigerella.
And-
It is a foram, but it's not Hastigerella digitata.
That's
Okay.
Solmissus.
All right,
It's-
Solmissus.
Move on.
Narcomedusa.
No, what happened to the
Narcomedusa.
giant blob?
It-
Did we lose it?
Yeah.
Yeah, we
I
didn't-
think
it
those
got
blobs are
swirled.
probably the most important ones.
Yeah,
if
it
you
was
see them
up in
again.
the hood basically. Of everything.
Wait, Manu. Can
No,
you
no,
say
no,
that
no, it's
again?
very important.
Can you say that on
Yeah,
video?
I'll say that on video. Those giant blobs that you see that are wriggling around, because we never see the cell membrane when when we catch them.
Mhm.
So this would be the only time. So Russ, if you ever see now a blobby looking thing,
Like that. I was gonna
But you were
collect
saying
that
it was the
before I got
most
distracted.
important
it's the
something.
most important thing in the world. In the whole world.
Said the
Blob
random-
against the other blob.
I think the
All right,
problem-
we can
bring
move
up the
on.
name, so we just let it
Yeah.
go.
Let's
like-
check that next.
Ah.
I'm sorry, Manu. I think we were gonna look at it a little bit more, but
So now we're going back to calling the thing that was a foraminifera and then a phaidarian or whatever back to being
Yeah,
a
no,
radio.
the glassy
Now
thing
we're,
we can call it
What is that?
Hastigirella foraminifera. Yeah,
so we
that's
can
a
call it
type
a
of
foram.
It
Okay.
is a foram. Hastigirella.
is that a copepod?
Is that,
That
a
is a
copepod?
that is a copepod, Dave. It's your day,
Zoom in.
I think, or is it? No, it could be a spiky. Could be a spiky risaria.
I do
Could
see
be a
two
risaria.
tentacles.
No,
I see way more than tentacles.
I don't know. I see two long things hanging from it.
It's two. It's the pair. If we want to get it, this is the one to
Oh,
get.
yeah, this,
It's a pair
Is this the
cylindrical
most important thing?
polydendrida.
I think the coincidence of you coming in here at this exact moment is really
what just
high.
happened?
You planted this, didn't
I
you?
did plant this.
What's-
Okay, let's
wait,
get a good picture
wait,
of this.
what's in the
It has a
back?
tentacle.
What's
Oh,
beyond?
there's something yellow, right? Did you
No,
see the
I'm
yellow
looking at a
puncture? Oh,
jelly
you're
that's
looking at a.
far
Okay.
further
Okay,
away.
can you just do a good video? of
Então, a
this?
gente está na discussão agora que esses organismos antigamente eram considerados
Let's start.
protozoários. E um protozoário não forma um grupo
So
monofilético,
somewhere
ou
on
seja,
the
não tem
vehicle there's a little
ancestral em
cage. He
comum exclusivo
released this
deles.
electronically. When he came
Eles
in.
foram subdivididos em vários
On the
grupos.
right.
stop your player around.
E essa agora está
This is-
classificada dentro de risária.
Good.
You sound like you're in pain, Manu.
No,
You know, you guys were like, when do people take the camera lens off? I want to take it off anytime you're just pacing through there. Just so like they can see the pacing.
Pain.
I know we're not gonna see it ever again in our lifetime. That's the pain.
It's there.
That's the
I'm
pain.
sure we'll see
This
it.
was our society's only chance of
What's this
meeting
little guy right
a
there?
Colodendria. No, focus, Alex. Focus.
Yeah,
Focus on the
It's not
two
Alex.
stars. There. Okay.
That's Mark.
Oh, that's
these are
Marco,
not the
yes.
droids we're looking
Yeah,
for.
this is the droid. Oh. Yeah.
I'm looking at the.
Okay,
flashing-
see, it
I'm
has
looking at
something. Oh yeah, I see.
The
I see.
flashing thing?
Okay, it has
Ah.
something inside its pouch, which would also be very strange for right there. Ah,
No, it's in
Uh, too close.
Human babies, it's abducted.
yeah.
Okay, how about we collect this for you? Would you
Yeah,
like it
let's
sucked
collect this one.
Does it
A
get
gente decidiu coletar esse agora.
Let's have.
D-sample, right? Or suction.
Yeah, no, always decent. Like no, the suction. No,
Suction,
never the.
it should be
the
robust enough for
infamous-
suction.
The suction.
Section 666.
Could you, 666, right?
No,
Down in
no,
the 666,
333.
where
No.
all organisms go to die.
Okay, it's on the right. It's on the right. Yeah, we can collect it
É,
actually.
isso é uma risária, é um bicho que a gente vai
Nope,
coletar
no, that's
agora.
not it.
Oh, that's not
Okay.
Apareceu outra coisa ali na câmera, mas não é ele.
They did a switcheroo. There we go.
There it is.
Oh, yes. We just rolled over 28 million annotations in Falkor. Like people might be multitasking, checking out the live stream.
É, e os risários são organismos unicelulados. Esse é um bicho bem grande. São dois, na verdade, que estão um do lado do outro.
I know what is.
Eles têm também um
Yeah, we have.
filamento.
We're 250K away from 5
Oh, this is like Bonifor
Esse vai ser
beyond that.
coletado.
What's a negative and people that delete?
It's
Focus,
it
basically
basically
says
says
it's
focus
not
on the right,
Oh.
right there.
But the positive annotations, we're coming up on 5 million. Let's see, before the end of this expedition. Oh.
Which one is it?
Pretty right.
And I promised to improve the consensus program.
Then I promise.
Yeah. Well, it takes multiple annotations now to.
We need a little autonomous drone we can fly out to collect these small things.
Yeah, that is the one. That is the one.
It's fewer than that.
Right there.
Screen.
Flying these roads based on
Do you want me to turn on the
your
laser
experience and all that.
to give you
So-
a different,
So it's a weighted.
a constrained
Yeah, so
perspective?
it could take 2 or 3 or even more votes sometimes to people disagree.
Yeah.
You know?
E agora o pessoal tá tentando posicionar aqui o ROV em
It is
frente ao,
small.
a esses risários
It's probably half a
para
centimeter,
poder
the
fazer a
what,
coleta. Ele vai ser
to a
coletado por
centimeter
sucção
tops.
através desse funil que vocês estão vendo na
So
tela.
after this, anything else you do on the benthos is going to seem like a walk in the park.
You're collecting
Right.
single cells, doesn't get smaller than this.
So,
Okay.
Things can hover over it for a while.
so if you put in, does putting in a negative actually slow down the whole process?
Nice.
More than it's worth?
Wow.
No, it can-
Wow. Okay.
It depends on the placement, whether the, I guess, sorry.
Is
Yes,
that
we
it?
got it. We got it.
Ah, ele
Close,
chegou
close,
lá,
close.
pegou, foi coletado.
We have it.
Maybe a
Okay,
smaller parking
rotate.
space type
Here we
of
go.
We do need smaller
We're gonna
buckets.
waste the
I think
space. We just need to get bigger things.
There's that. Or collect more of them. You can collect hundreds of them.
We'll just
Right,
keep
just
in
pack it
mind
full.
the number and we'll
Yeah.
come back to it
Well,
and stuff.
We could
Fill it
load a
all
whole
with that
dive up
stuff.
with Protosin and the rest of us get to go to bed.
I mean, that's not a bad idea.
yeah. Going to bed is also not a bad idea.
Leave him to his own
Okay,
madness.
I think we have it. We have it. So now, as long as it doesn't get flushed out.
Yeah, that's the problem, is they're just so small.
They are so small.
We've got the smaller mesh on these things, but still. Kind of don't have to both be in here. With here.
I mean, I'll probably sit
Neither
here
will all
anyway.
angling Manu, it's fun.
Yeah, you should hang out.
There was a big jolly, I think.
There was something creeping around in the back.
Yeah.
No doubt, just
You know,
beyond the
this
light.
pair
there's a
thing
ton more.
is turning out to be more common.
This,
We've now seen these pairs, potentially 3 different species of variants. That
Yeah, we
they-
saw a couple
they
of
live in
singles,
pairs,
but there've been a lot
but
of
the
pairs.
only way you can be a pair is if you do it during cell division, which would be kind of clonally, they're genetically very close. But somehow it's useful to be in pairs.
Yeah, or
I mean, it's
we're
not
catching
so-
them,
Ah,
that
yeah.
the division
Once a day,
process
né?
takes so long that the bottleneck is there, and so when you see
That could
them,
be.
it's likely that they're the ones. It's just a,
No, but the division.
aliasing
No,
problem.
division doesn't. You
It's,
don't need
you-
to mineralize.
it's hard to take a
have the
silica
division.
skeleton apart.
They grow the skeleton.
That was
They-
that was
Yeah.
a whole
But to
but it
keep
still might be
kilo,
the,
whatever.
keep it that
kilo
long.
the bottleneck.
I
I
don't think
mean,
there's a some genetic
All right,
trick that they're playing.
let's see what that is if we can stop on it.
Maybe one of them end up being.
Because this is, here we go, there's our second shot at it.
What is that?
Is that right area, or is, I don't think it's a jelly, it's not moving.
Captura?
Most
Could
of
be
the
blue
luz
leaf
que
blubber.
Yeah, that
dele.
looks like a
Aí vai aparecer uma
larvation.
bolinha.
Yeah.
years ago I did.
You've seen
It's a
blubber?
really
Like a
collapsed
common
Larbacean
Yeah.
house.
This,
Super
Robin Williams.
small.
yeah.
Aí você dá 2 cliques em cima.
That is a larvation house.
É
Yeah.
aqui.
Oh,
I think
It's,
so.
it's awfully collapsed to be a Larbacean house, it's something different.
I don't know.
Entendeu os nomes.
Okay, I'm wrong. You're right, it's collapsed Larbacean house.
É fácil, mas...
It's just goofed up.
It could be a fritillarid. They
Is
can
it
collapse. It is a
fritillaria?
fritillarid house. The fritillaria is
you're right.
actually there
It is there.
because they can swim the house around.
Oh, alright.
Let's watch it.
I think it's there.
I think it's there.
Ah, será outro recordado, né, que secreta
It might.
He's
a
there.
túnica.
Yeah, he's there, definitely.
He's still
Aí
there.
o dono da túnica tá em casa, assim, às vezes ele sai, abandona, vai embora.
Let's desample it because he's holding on to it really well.
Oh. Just watch.
He's out.
Okay, it's no
Ah,
longer
saiu,
there.
dá pra
We've
ver ele
knocked him
saindo
out of the
da
house.
túnica
Okay,
ali.
but it was a fritillaria.
Yeah, I think you're right.
Fritillaria.
F-R-I-T-T.
All the other things they have to do.
Yeah, go
Feels so
forward slowly. We're
coarse
gonna look for
after
another
this.
house.
Like, we're looking at some of the most delicate things, the most delicate structures.
Hopefully we'll see one a little farther away when we're more under
Well, and
control.
all the ones we're looking at
Let's
right now
check
are
these
all
two things out. One is
And by the
probably a
time they
keating
get
nap
to,
or a
like,
fish.
Is that a fish?
size on the screen,
Fish.
inside that hood.
Fish.
Up
fish
there.
Cyclothony?
Yeah, cyclothony.
And if you try to pull away from them, you get all the vortices around the edge of the hood, and it just throws them.
Ciclotone é peixe da ordem Stomiformes.
Is this the same thing we were looking at, or is this
Esse é
another
o,
one?
grupo de vertebrados mais abundante do mundo. Tem
So how come?
o equivalente ao crio dos peixes, eles são
Because I catch them at
planctófagos,
this
né?
step.
That's a lot.
I haven't
That's
seen
a
single
lot.
egg.
Here's another colony for you.
Oh. It's a Tuscarora. No. This is
No, it's not a Tuscarora. There's no sphere left.
Whoa, whoa, whoa.
But it's
Zoom
like
in.
it's like 6 or 7 of them.
Nossa, que legal. Esse
It's
teu
another
rosário.
colonial
Então,
Beldarian.
Yeah.
grupinho ali.
But let's see.
Dougal's paying me, $50. He said if I can load up with produce so he can go to bed.
You should ask for more money.
I probably.
I will pay you even more.
Okay.
I'll pay you
It's-
more than what
It's
Dougal is
now
paying
we're
you.
into a bidding war.
To do the same thing, which Russ, I think that's great.
Oh, that's what he said? Yeah, we should load this up.
Okay, go ahead.
This would be
Go ahead and take it.
something new. This is something we've not seen before, at least in this group.
You said Theodarian?
Never
There.
seen with human eyes at this depth before.
In this place.
Yeah.
In this
You leave
place.
those canises free for Tina boys. What are you talking about?
No, this is very
Isso é
special.
mais uma risária aqui, é um bicho colonial,
I leave the room for a
aparentemente.
few minutes and look what happens.
Time to show the video.
You will be collecting
Agora os pescadores estão aqui no argumento se deveriam coletar ou deixar isso livre para outros organismos.
Suddenly we're all part of Club Rhizarian instead of Club Siphonophore.
Okay, I promise. This one. This one.
Just this one?
Just
Wait.
for this time. It's for this time.
Just
Let me
this one.
correct
So wait a
myself.
minute. Record that. Record that.
Yeah.
Say that again.
Just this one. This is too special.
Is this a produce noticed?
This is
It's a Tuscadarian, but it's not
It's-
a Tuscadarian.
just put it in bucket 3.
It looks like a colony.
Two.
it is. It's about 6 or 7 individuals in
How
it.
do I feel
Yeah,
about
it's so
proteus
sexual.
versus Tinaphores? Come on.
Oh, where did it go?
Let's suck. You're good to suck it?
You have an eye? No.
We did pretty well last time.
It's okay if you miss though, because there's Tinaphores
No.
waiting for that section.
What are we doing?
It's that one.
Russ, you've lost control.
Yeah, we should be in sampler.
Pound that little finger on that desk.
Oh, well, we should be in sampler 4 as the next open sampler. Yep.
This one.
Sampler 4 we should be in.
Yes.
Manu, professor Manu Prokаш, ele é professor da Universidade de
Yes.
Stanford, na Califórnia, e ele está super empolgado com esse bicho, esse organismo. Né? Na verdade não é um bicho, é um risário, é um organismo unicelular,
This is
e aí tem vários organismos formando uma pequena colônia.
Yeah.
Or seal it, the pair.
Aren't we gonna flush them?
No, it would just do a lot of damage if we leave it running for the whole collection process of another animal. You only do that with something really robust.
E toda essa empolgação do professor Manu Prokаш está
Plus, if these are
divertindo todo
different,
mundo
you don't
aqui.
want to mix them up. If they're not different, you
No,
have,
they're absolutely different for sure.
well, they're little. And a
Passando um
suggested
camarãozinho
shrimp
ali, ó.
going behind it, the long antennae. Probably going to try and eat the rhizaria if we don't collect it fast enough.
Whoa. Is there already something in 4?
No.
No.
Yeah, well, there was a lot of movement that was happening in there.
Como assim separar?
Okay. All right, thank you.
mas...
You guys just see that?
I didn't.
Yeah.
Did you see
Yeah.
it?
Yeah,
Yep.
it's
Oh
right
my
there.
god.
They're
Four
everywhere.
of the same bugs that we are trying to collect. So this
Yeah.
depth is very specific.
I mean, but you said
Yeah,
that
no,
was the
that-
one.
That's the one. That's the one.
Either that or
We
the,
even have
what
a
we were looking
recording.
for is what we just saw.
Oh,
Down to
yes,
2. Good
yes,
job.
Yes. Okay, we got it.
Okay. Keep
You
going.
could stack
got it.
them.
There it
Okay.
is.
That's
See
it.
it.
Yeah.
Yeah,
You could
chegou.
stack them.
How come the bucket's working
the
so
same
quick
bucket.
now?
This is what we do with the obvious.
They
Oh,
were-
we
but
took
I think
Dougal's
it was
thing
such
out.
a
Yeah, you took the
abundance in
shadow
such a
graph off.
small
You could
place.
catch
Oh,
them.
we need to leave it out then,
Stack
Dougal. It
them
works
and put
much
them
better
in a
without it.
container.
That's a good idea. Actually, yeah, no, let's just take it what we have. No, but what's in the bucket will then see the flux from the suction, right?
Yeah, but not a lot.
But
What
it
do you
cannot
What do you mean, if you
escape.
go
That
back,
was
if you go
once a
back and put it in?
unicorn.
We'll stack them
We
all
can
the
try.
time. You just do slow pumps. And they're not like they're not fast. So
This marine organism is going to get all this water shot at it.
that's what I would
Right,
do.
all this salt
if
water.
I were you.
Ooh.
Yeah.
No. No.
Do it, Manu.
No, don't. Get
Yeah,
me wet.
see, right there. Is that the same? No.
Okay, we're looking for one of those houses.
How
those
many
kind
there
of-
are?
In this particular place at that particular moment.
So let's keep going slower so we have a chance of stopping
No,
when we
we've
find
seen them.
one.
They,
A gente está aqui, o ROV SuBastian
Não,
agora
mas
está a 520 metros de profundidade, 523 metros de profundidade.
We didn't
E
even
essa
film
parece
it.
a
We turned the
profundidade
camera
ideal
off
You
para
as
have
we were
to
going to do it.
understand, it's like these things don't school, there's no
Another,
like
Kelly
it really
Cochran.
is strange to see abundance at a particular depth.
Yeah, is there a density
It's-
climb
Keep
here?
going.
We,
Yeah,
Where
what
they would-
information is here?
where they would.
Try down
No,
here.
we're too deep. 500, yeah, we're way too deep. Oh, it's at the base of
No,
another
it's
dial.
not.
But this
After-
is not dial. I mean, their cell
That's
cycle,
a
we don't even
fish
know.
again.
This is, my guess is
Um,
Tem peixe ali, não
For their
sei exatamente o que é.
some
Which,
of the minerals
which
that they're
would then
making.
explain why you would see it if it's a bottleneck for the division,
Yeah.
right? I mean, you see the same thing in larval development, where you see an abundance of a certain stage, and it's because
Slow
they're
down.
fast to get there and slow to get out.
Yeah, yeah. I mean, the mineral sequestration is
no,
what's
that's
slow
just
because you have to sequester atom by atom to get that
don't
particular
tell her
mineral.
that's another rise area.
Yeah.
So, like the storm shim one,
Keep going.
the concentration of storm shim in the ocean is so low, and yet this is
Is that what
There's
they're
one
collecting
of
to
the-
lay
one of the
down?
colonial
That's
ones
Aquarium
right.
is laid down. Yeah, storm shim.
Oh.
But it's the only like they have a molecular machine. They're the only ones that
That
can
we're-
sequester,
Yeah.
and we know nothing else. Like when you measure the native concentration, it's insane. So every atom is somehow being sequestered.
Huh.
And then the moment they die, the concentration outside is so low, it goes in reverse and they dissolve. So nobody can fix them. There's no historic record. There's no fossil record
Yeah.
because of
Funny.
the concentration
Right.
gradient
Up
is
here.
so strong. So the
Slow
only way we
down.
keep them, the fossils in the lab, is spike
It's a
a ton
house.
of
There's an
super
animal
expensive
in it.
storm ship,
Right.
so that the
So let's try and
chemistry
set up.
doesn't reverse
Make
back.
sure to have a zoom in on the
Yeah,
animal.
that's
Then
good.
we'll
Has
do the
anybody
collect-
broke the life cycle and can rear them?
Oh, nothing.
No.
It's
It-
in too close,
never
it just
been
failed.
cultured. No cell division seen. Nothing. I mean, we
Yeah.
barely know how many nuclei they have. Like no live imaging ever been done. All
The
fossil.
that
ocean
wasn't
just started
like a
with the
blur in
number that we
the
have
house.
now. And anytime we collect one, it's one fewer.
Yeah, you're right about that.
We're draining them.
We
They
are
have no
probably
life
draining
cycle.
them.
They
How-
just exist
yeah,
in
big
this
is-
limited quantity.
Yeah, but you know, imagine if these organisms indeed are 100 years old. Like, what does that mean?
It means they're-
Like,
I mean, they find bacteria.
like, what is the fundamental limit of a cell cycle? Right? I mean, the bacteria, yeah, the
Yeah.
methanogens they
Yeah,
have
yeah.
are
And the
like
ones that-
dividing once in 2 months, 3 months.
Yeah, the ones in ice.
Yeah.
I mean, those could be 10,000 years.
Yeah.
I mean, they accumulate the growth rates of them, or estimate the growth rates.
That
Right.
is beautiful. Another rise area. We have hit a rise area.
Mais uma risária ali.
Oh my goodness.
O professor Manu Procash vai encher
It's a great time to come in.
uns baldinhos de risária.
What? Yeah, you can see that.
El
So
ojo.
the point around this is that the membrane is inflated so much collect it.
Nice shirt. Look at that.
Thanks.
You are in party mode.
Yeah, it's party mode.
It was
but
perfectly
it's hump day.
spherical.
Yeah.
Yeah,
See
it got hit
how
by the
deformable it
wash.
is?
Yeah, yeah.
Yeah.
Oh, it's dinner time.
Okay,
though.
It's going to be
Zoom in
really
on it.
fragile to
Yeah.
try and collect, so
Sort of
we're not going to have much luck with
Yeah,
it.
but let's get really good shot.
Did you not happy with that?
I think there was one we were just about to be in focus. Let me see if we can
Breakfast
see the
actually.
core.
Should just be back there. It
right
is.
I've just missed
Is it a
lunch.
single core? Instead of-
Although I guess not a single, so this will be.
Yeah, what did we have?
Yeah,
It's,
what do you do?
you know,
What do you have?
it's literally we are
I was
looking
gonna go
at
lunch, but you
a
can go
soap
eat
bubble.
first and I'll sit in your
Quite
chair.
literally, because
Sit
all
in
of
this
that
chair.
was just a
Sit in
membrane,
this chair.
and it was behaving like a soap bubble,
Yeah.
See if there's something
except
really cool.
underwater at this high pressure.
We've hit quite a few good things already, so.
Okay,
see if
cool.
we
There
can
it is,
do
there it is,
for us to
there it
jump
is.
on it.
That it still centered?
Yeah.
How big do you think this is, Russ?
You saw that copepod, Russ?
I was focused on trying to keep track of this.
I mean, it's pretty small. You can see it.
I suspect a lot of these are
Yeah.
Oh, I
copepods,
see
these
it.
specks.
Oh no, but one came into pretty good focus.
No, I missed it.
It's clear to me whether the central capsule is central or is this all periphery? The chain. Oh yeah, there is the central capsule. It's there. Oh, that's a good shot. Nice.
Okay, inside.
Okay, I think that was really good.
Yeah.
So,
In the hood.
oh, now it just goes
Yeah, yeah. So when it goes
past
like
the
just
light.
completely invisible, yeah, it's absorbed inside the hood
And
at
once
that
you get
point.
over the lighting field.
There it is.
Oh.
Made it out. Goes. Nice.
Did we squiddle it? What, did you squiddle that?
I don't hear.
Sorry, did you squiddle that one? The blob? Yeah.
Sorry?
The blob?
Yeah,
you
yeah, yeah.
squiddle it? Yeah. What did you put
Slowly.
it as? Yeah.
His area.
Right there, yeah.
His area.
If this doesn't pan out, we'll go for the target up above it. This is probably not what we want. Let's look back there.
Right now.
It's a soup down here?
No,
Yeah.
drop it on
One,
both.
The
one
number
every
of
10,000
Another one,
cubic
look at
It's
meters.
really
I know.
creepy, creeping speed.
This is super exciting. I mean, it teaches us something about abundance. In the,
I think it's amazing that the oxygen concentration here is only 36.
yeah.
That is
Yeah.
extraordinary.
Yeah.
What is consuming all of that oxygen?
You know, the oxygen minimum zones are the biggest mystery right now. They're
Yeah.
growing, and they're deep, but we don't know why.
Yeah,
It
what's-
doesn't make sense.
I mean, I guess
Why
warming,
is it-
it's 7 degrees. Which is, I mean,
Yeah.
We're at
that's the
530
surface
meters.
ocean in
Oh, it's
Maine.
7
Yeah.
degrees?
Yeah,
Yeah.
Wow.
We're at 7
Which is
to
hot.
7 meters.
It is hot
No,
for
We're
that.
keep going.
yeah.
That's crazy.
That's why
Yeah,
we have
there's
the
some-
soup.
So much, right?
Yeah.
It's warm soup.
Can't have
It's
cold
Is it right there?
soup.
Yeah, it's a warm soup.
Yeah, it's a warm soup. Oh. What is that?
No gazpacho.
No gazpacho.
Right,
That's right.
There's another thread-like thing at the back.
All this soup
There,
and
maybe.
gazpacho is making me hungry. I might have to go eat
Wonder
my
what it
midnight
is.
meal.
Chat, if you can believe, it was rib night last
Keep going
night.
forward slowly.
Yeah,
That was
Ribs?
wild.
I think
Oh,
I
it's
had.
time.
We were out here eating ribs in the
Yeah,
middle
it's
of the
time.
night.
I think I had the meat sweats last night.
I went to sleep so
Who
far
eats?
after that it didn't present an issue.
Meat sweats.
Meat sweats.
What are those?
I'm not sure how to know.
When you have 52 feet of intestine full of red meat,
And then you go to sleep and you sweat it out.
you sweat.
Oh, you
Over
sweat.
here,
Meat sweat, I get it.
that looks like it's a house.
No fish.
No,
Where? Right
Oh
there?
no, it's a shrimp.
Oh,
It's a
it's
copepod. Yeah.
It's sinking. It's
It's
sinking.
a-
Oh.
this,
Oh, there's a Medusa
Amphipod.
down there. No, it is a shrimp, no?
What?
What is that? No, not a shrimp, a copepod? Did you see how it was sinking first and then
Okay,
started
just
swimming?
keep trying.
Yeah.
You see it swimming,
Yeah,
and
it
then
was
just starts
oriented
sinking.
down. Now it's swimming. You can
Here,
tell.
up here?
Oh, that?
That's an amphipod.
That's an amphipod.
Amphipod. How about the Medusa?
Oh, and we have our
Oh, there's your
Colid
rod, Brad.
Rica. Yeah, another. Oh, that's beautiful. That is beautiful. Have one of those. Oh, wow. Actually, this is useful because we finally get the right orientation of swimming for them.
Oh.
Wow,
That's
that's
nice.
gonna make Karen happy.
We can collect.
Karen, can you
We
hear
can
us?
collect it. We
Karen.
should suck that thing up. Actually, that would be
Nice.
really good.
Okay, can you keep
Oh, it's
following
giant.
us?
Did you
Oh.
see it's giant
That was a
eyeball?
classic
That was a
dive.
really good-
That
There it
was
is.
a classic dive.
That was
when
really
they
good, you
are
guys.
fresh.
That is
That is
huge
really
eyeballs.
good.
This
Data.
is an
Pode
amphipod.
partir?
Como
Do
que
you
chama
see
isso
its
aqui?
eyes?
The other was an archomedusae.
Enormous.
Wow.
Oh,
Wow.
it's like
Yeah,
a
do this in the
cyclops.
lab. We're trying to
Bicho
do this
do-
in the lab.
You need a really big lab.
That is really beautiful.
Agora é um momento bom
Wow.
pra parar.
Oh, and then it does a dive.
I love the little tail tuck. That's a good one.
Tail
Yes.
tuck?
Yeah. And they have
But
latches
this is
for
not
those
copper.
tails.
It's not.
Yeah, and they're straight up. They have a little seat.
Todo seu.
And you
Sorry.
can see the eye. You see the transparent
Então,
eye.
aperto aí,
Aperta aqui.
aperto,
Okay,
anotei
let's leave the
marco o
amphipod.
pontinho
Pontinhos, anota o nome
e o nome.
e fecha com janela.
E fecha onde?
Should we
Vai,
collect this
faz
one?
Muy bien.
Are we
We
able
will
Ah,
to?
never be able to collect that.
Oh, it's too fast?
It'll be too erratic and too fast.
How big was that?
Bom,
Centimeter
faço um
tops.
aleatório, assim, não precisa salvar ou salva raciocínio.
Não, sempre
That
salva.
balance
Não
Ah,
was
tá.
amazing.
great.
Você pode apagar o ponto.
Balance
It's really
is
beautiful.
Ah,
Outra
tá.
trabalho.
And then look at that shot.
Jelly
We both
over
are
there.
friends. Kind
Oh,
of.
here.
Oh, tá.
We got a rice area
Yeah.
and a
Rice
Oh,
area.
and
It's a silo.
Não.
It's a
Ah,
lonely
Fica
ghost
aqui.
down there. Maybe they
Anotei.
were friends.
Clica duas vezes.
Cross
area.
species.
Silo.
Aí,
Cross
Yeah,
phyla.
this is the
Escreve aí o nome.
protozoans.
Area should be
Se você
Cross
Mas não,
Fiodarium.
qual que é o nome?
There's so
Eh,
few
What's the
cross
name of
kingdom
this animal? Animal organism?
Colidendrica.
Com...
Oh, there is a ctenophore lobate.
Yeah, little one.
Yeah.
So cute.
So
Is not
right there.
recognizing?
Yeah, sorry.
It is
Just usually go here.
That's
Right
a,
area.
Ah,
is this like the one we
Circuzoa.
collected the other day?
Endozoa, no. Ranophacilla.
Big name.
Yeah. I have it in here.
Oh, in sweets, every. No?
Yeah.
Oh no, not
It
another
should be in
one.
here. Okay, we'll just put it for right now.
oh.
That is a
Right there.
Praedae, a Calicopteran,
Ah, tá.
Siphonophore. or
Did it
is
get labeled?
even
No.
No. Yeah.
Yeah, it got
Yes.
labeled. Yeah. Okay, it has it.
I think it's a head off of a Praed.
What's the two masses in there? The
They're just the viscera,
Oh,
tentacles.
okay.
Siphonophore.
Se você vier aqui, ó. Olha aqui. Se vier aqui, vai ter os últimos.
Yeah, when you go to the past, you can always see and find it. Siphon.
The last 20 or 50.
Siphonophore. Do we know something else about this?
I just put like big name, big groups.
Ah, okay.
Just do a frame grab on that.
Yeah, this
Right.
room is the most
Exactly.
Before you
amazing
get to the
room.
next screen.
You'll hear the screaming, but you won't be able to get here in time.
Okay.
I often try to in the lab. I it's like not really.
Aqui?
I think there's something going
Good
on
thing
in
we're not
there.
rougher seas because
No.
you tumble down the stairs.
Você vier aqui no botãozinho.
I
você
wonder
clica no
if that
direito,
impacts
Ah.
the
você
piloting
clica,
deep down.
Ah,
as well.
tá. Tá
Whether
bem.
the tether has enough connectivity.
fecha.
Oh, I think they say they
They
leave it
wave it
loose.
first.
Yeah, I know, but still,
Tá.
like your balance, there must be an impact
Tá.
on their
It's a bigger deal for
maneuverability.
you.
Vou sobreviver.
Yeah.
Vamos botar então, maybe we could ask
Sim,
them
sim.
what perspective.
Mas agora eu fico aqui.
Totally just
Two,
like on
with
smack
that one and one up there.
Have you seen
Oh,
That's right. I totally know what's going on. Let me
Oh, that one.
Oh, what was
a
that black thing?
non-diver explain it to you.
where?
It was a black thing that passed
That's
by.
really
No.
fast.
Yeah, but it's super fast.
Dang it.
Ultra bad camouflage.
Yeah.
That's the stuff I want to
The
see.
ghost of the ghost of the Risaria. Oh my goodness. Look at
That's
this.
how we saw the gopher eel yesterday.
Oh, the
had a
gulper was
teeniest
cool.
little
The
animal.
Keeper by the other
gulper
side.
was
So fast. and
cool.
I found it.
Whose watch was it? In your watch?
Yeah. That was a special treat.
I bet they feed on.
Oh, look
They
at that.
probably they eat everything. Yeah. Actually, I just found a paper today morning that
Today.
Astigerella keeps
And
Pyrocystis
another
as
Thalassophilus.
a dinoflagellate.
What?
For what?
T4.
For in the
You
harvest
No.
mean for
of
pets?
That's not clear. It doesn't engulf it, but it has very particular association, like as decorations maybe, because
Oh.
Pyrocystis is bioluminescent.
Yep, keep going on.
as
So
decoration.
as decoration, like you are like a tree,
Yeah.
Those were
like the glass. It's what is it called? It's punk or is not good enough, so it's gonna do more. Like what is when you do the car? When you jazz up your car, what is it called?
Trick it out.
No, another term. Yeah, tricky our car out.
Pump my ride.
detailing.
Or pimp my ride.
Yeah,
That's what I
yeah,
always have.
That's what it's doing. It's pimping. It's
yeah, it's
Better off.
pimping its ride by Pyrocystis. Like as if all this glass dazzle is not enough. Let me decorate it. It's insane. And it's not eating them. It's
And it's
not
not
like it
eating
can
them.
have between 5 to 70 cells,
But it could be
but only one species.
could be taking some of its byproduct.
It could be taking a byproduct.
Yeah.
That would be cool.
Yeah, that'd
Yeah.
be cool.
Yeah.
Yeah, that kind of mutualism or something.
Yeah. But your copepods, it's eating all of them.
It's,
I have so many
Ooh,
deep
that's what
water
we're looking
copepods
for.
only because of the
It will
rhizaria.
probably
Oh.
be.
oh,
yeah.
so you have them. You're still keeping them.
Yeah, I'm
This
I'm
one?
keeping all
Yeah,
the
it's
rhizaria
as
alive.
elevation.
because
Oh.
I'm
Can you go
trying
a
to
couple frames back?
do stains
Or
and
can you not do that?
watch what happens.
No.
But
Here?
the
No.
tentacles,
It was, you
I
can mark it as a
see.
house.
Oh.
Okay.
So that would be a good collection
There's a
method
good, yeah,
for
More
copepods.
produce.
feeding them that. You know, the one thing that we haven't been able to.
Oh,
bring to
that's
life-
beautiful. Oh,
another pair.
another pair. Can we just video that? Just so we can watch for that one to see how they are connected.
It's like protozoan bachelorette. They're all paired out.
They have, don't they have a connection website?
Connection website.
What is that now? Let's focus. What is that? Any guesses? It's,
But
it
thing in
could be
front of us?
Risaria. Oh yeah. Oh, no.
No, that might be a
No.
small colony of.
Yeah. No, it's not a
It's only
Risaria. It's
opposite.
quite small.
But it is. in the
I'm
wash.
lost. Can I write just house? No.
Do miscellaneous. Yeah, we have the category for miscellaneous in the very end. Oh,
Living other.
No.
that? I don't know what that ghost-like thing is in your screen. You can call it Casper. Now, what's the ghost in Ghostbusters? It's made of
Marshmallow Man or something.
Marshmallow Man, yeah. Yeah, that isn't that what it looks like? Sorry, yeah, if you go in, I'll show you where the other is. Right here, if you let this go first, and then down below. Oh,
No, I didn't mark the point.
yeah, annotate. I don't know what to do now.
No.
Unmask coded
I vote
text.
for
That's it.
Okay.
Yeah, I mean,
Not sure what jelly that was.
oh, this room is
Send him, send him away.
mistake.
That was.
should have eaten first.
That was a mistake. I can't believe he's gonna be gone all week now.
So Mrs.
Shame to this chair.
out here.
All right. I'll go get something to eat and I'll send him down.
Oh, Emily. Can we have you for pictures? Or you're in charge today?
Yeah, well,
Okay,
no, I'll be out soon.
Oh, it bailed already. Slow down, coming
I'm
out
gonna
slow.
And then I promised Karen I'd do the tomatoes next because I did a bunch of
Oh,
those.
no, you have
But
like
then
some-
after that, I can do more.
It's like
Okay.
deals going
Okay,
on.
yeah.
This is like
Yeah.
Karen's been waiting.
April,
for me.
April 26th.
I mean, Russ is
Yeah.
taking some calls, so Dougal and I are fighting over it.
Yeah.
So yeah,
But
your machine
I'm
can
sure
also.
Yost will be your Senate.
Okay,
Okay.
sorry.
No, it's okay. I'm sorry we had a delay
so
today.
my
It's out of
proposal
commission.
would be, let's image a little bit more
Yeah,
of
let's
that.
zoom in on it a bit.
No, I
For
don't know.
stable.
Mark that one.
Yes,
Larvation is
I
bad.
believe it's a fritillaria house again.
it's larvation. We'll see here.
Fritillarians are the ones that can fold up like origami, right?
Well, the house collapses and the walls don't stick together. They can blow them back up.
That's so cool.
Not seeing the animal yet, though. Let's just give it a second to see if we see any change in it. I'm not seeing it on the outside like we did in the other case. We could kind of see the animal on the outside. Wait, that might be it. Try and get it in focus. Definitely a fritillaria house. That could be the animal. Yep, the animal was there. It's right there now. Here. Slow down. It's another house. Oh, and a sparkly risaria and a little wee.
Can I capture
Medusa.
another picture before labeling?
Okay, this animal is there
Yeah, 'cause
based
there's
on that
also
white
now
spot.
that-
If you can hold back and just zoom, okay, we want to collect this with the D sampler.
No. Shall we do a DPIV?
We will never have it survive that process.
Okay.
Because I'm not finding larvarian here.
It's got something stuck to it, huh? Oh.
I didn't forget, we had a heated discussion, but Dave keeps telling me that you were
Okay, the animal has just come out.
a mere shell of your former self.
So I don't think that's going to work unless you think you can get that into a D sampler.
I'm gonna eat.
You will knock yourself out.
But,
Okay, here we go. Do we want one of these paired? Right now, we got the vehicle under control. Just come up very slightly. There we go. You think we can suck that up?
Yeah.
All right, let's suck it up. We get a nice view of the house.
let's keep the suction gentle on this because the pairs break up until it shows up.
Yeah, so responding to the chat, the Larvasian houses are built for the animal to be able to abandon them. They eventually get clogged with food, and they have to replace them with another one. The animal typically has several of them stacked up against its body for reinflating when the one is cast off. So when they get alarmed or when the animal house starts clogging, they abandon the old house. And the houses contribute to the marine snow pool.
No,
I don't think we got it.
nothing in there.
No, I think
Is that the pair still here, right?
No, that is.
Oh, that's a
the
jelly, sorry.
No. This is a foram, the Fastigerella. But the pair, yeah, just have this go in anyway. Oh. You can load this in the same computer, just like the thing on summer. Awesome.
Well, you got that instead.
Yeah, that one we didn't get.
Yeah, we didn't get that. But I really thought that it was in.
No, it was.
There we go, it's in.
Sorry.
Okay, shut the sampler down. It's actually a good sized one based on the amount of light reflecting off it.
Oh, it's fast into the path because yeah. Down the
Which
path.
one is
Got
it?
it.
6. It's
Okay, so 5 has Fastigerella. Fastigerella. It's a Rhizaria foram.
okay.
We don't know what species it is.
So this, but we just sampled into 6, right?
5.
Oh, we sampled into 5. Okay, I'm
Five,
getting.
I think 5.
Bucket five.
All right.
Okay, you're in charge of the Rhizarian soup.
Okay.
For some idiots.
We have tried several Larvasian houses and we keep dusting them.
Okay.
We have it numbered up with, loaded up with 3 different produce, so.
Okay.
We got our fill of produce for the venue.
Yeah, probably. Sounds good.
We have Hortensia, I think, in one. What else do we have on there?
Checkers.
Yeah. Yeah, we've got 3 in a row that are area.
Okay.
Better notes are
sounds good.
right beside you.
Okay.
So 5 is, 6 is the
Okay.
next one.
Okay. Cool. Sounds good. The soup is
It's-
super good.
Okay, it might be that the Larvasians are a little bit more timed up here.
Where are we at?
I think they were.
What are you doing,
down
Tyler?
540.
About 5, I think 3:15.
Okay.
So,
Stop as well? Our waiters back on.
Okay.
You're headed to 6, yeah?
5 buckets left, or
Okay.
3 buckets left.
Yeah.
Both
Sounds
the
good.
samples are available.
Yeah. And no Niskins on this library?
No Niskins on the stuff.
No Niskins.
Hey, cool.
And
Hey.
when I came in there, the outfit was gone.
Hey, Chloe. How are you?
Those guys.
Hey, everybody. Karen Osborne signing on here.
And Barry had some problems there. Equipment.
We are at about
Everybody was
5:50.
green, and then power on, and then they had a telemetry problem with the iris, I think.
Yeah.
Zero.
Probably rosacea. No. Kind of tempting, but Oh, the for Nemo.
Yeah, so you heard.
If there's a chance you
They
could
go
go see this guy. Might be a larvation, I think.
Right there, you go.
Get a close-up look at it and then try to put it in a D would be optimal. It's probably too small to scan. It's very small. Let's see any movement.
Don't blame you. Just on the ice. Yeah. Tyler, don't you cannot can't believe that they have
He's in detention after this, trust me.
his eyes. Everybody. Don't blame me.
He's doing
Which
a
is a little-
laboratory.
Yes.
I don't see an animal in there. We can leave it.
Because I
Yeah.
do something.
You did something.
Yeah, I don't know why.
Nice when they have some color, something
Yeah.
to them, so you can see them
Ah.
Yeah,
when they drop their houses and swim away. No chance. So that specimen we got on the last dive.
Yeah.
So nice.
Got top end.
these those blue cameras are off. Put a front plate back
Turn
on.
heading down, head
Still
up.
light.
Down. We're kind of hunting down to 600.
Okay, we should have a
And then
big
we'll
camera, so-
head back up.
Oh,
Once
yeah.
more. Ah, okay.
You take the shit out of
Yeah.
those, a couple.
I said. Okay. Full speed ahead. No, I don't think so, but let's look at these guys to the left. I don't believe
Okay.
holding hands.
All right, then, Eric. We're gonna put these in a D sampler. Papa Shrimp, who keeps wanting to be on camera.
4 and 5? Yeah.
Yeah.
I'm gonna try this first.
Yep, make sure you know which one you're going in.
Oh, I'm coming close.
If you just release it, it'll go back to something.
Alright. I'm gonna trim down a bit, let you get a bucket.
Got it?
So do you know if these are the same as the ones
Got
they
it.
got before?
No,
I don't know which ones they did.
Walking over.
I'm not sure either. That way it was these.
There you go.
Oh, okay. Probably the same, but
Come up, nice and
it's all right.
Yeah, with it.
It's in.
Mm.
Nice. Ready?
All right.
No
That's for the
second.
night then.
No second try needed. That was D1, yeah.
Yeah.
All right. Let's find some Tomopteris. Another protease forward. Like it's not even there.
You ain't even there.
Kind of a dead spot according to the EK80.
Where is he?
I think this was collected on his behalf.
Other than the Alciopids of the surface and the Tomopteris, we haven't seen a single other polychaete. No Lepidorhynchids, no Piovius, which we don't really expect here, but there is one species in the North Atlantic. Kind of would have expected one down here, but not a one.
See you inside.
It is super weird.
That's the EK80 been pretty good predicting where to go.
You know, everything runs away from us.
Are you getting a lot of good stuff?
We can go in those nice thick layers, but they disperse as soon as we get there. You can actually see it, right? Like you see this like tight thing, and we go there, and suddenly it becomes like, and that's a huge scale, right?
Yeah.
So
Disruptive force.
we are definitely a disruptive force. Can we look at that white spot over there?
Oh, okay.
Yeah, they never do.
Yeah,
This one is just the same.
Yeah.
on the drive. 6.
No. What up, man?
We should be up by. We should be up by 2:30.
Yep.
Karen, have you eaten dinner? Yeah, you did.
I did.
I saw you.
Yeah. Yeah. What is that? Kind of almost looks like another polychaete we haven't seen yet. Strangely. Like a Pelagobia. Quite possibly a Pelagobia. It's very small, isn't it?
Yeah, I can't even see it on the. Yeah, I think.
Yeah, I think it's like almost in the camera. Oh, that's definitely a Pelagobia. They're not the most
What's that,
coordinated
Karen?
things in the
Yeah.
world.
Oh,
Pelagobia?
Clumsy looking.
it's a worm.
Yeah, it's a, sorry. Annelid, Lepidorhynchidae, Pelagobia. You think you can suck that up?
Try.
It's pretty tiny.
See
I'm
it
afraid
in
if we get
this.
too far away from it, we will. Oh my god, it's really tiny.
Cadê
essa
merda?
Yeah.
Here.
Are you happy on the jar we've got?
Wow.
Jar we've got on 6.
We're on 6. 6 is fine. Nothing in there.
We're gonna try to hoover this guy up. This 3 pixel wide thing. Can you give me a little bit of leash too, Leah?
Oh, he went down. Down to the right off the science cam. I haven't seen a single one of these guys at the toes. Do you still see him? One of many tiny specks. Super long shot, and he's barely bigger than marine snow, and smaller than some marine snow. They are something that if we stop, they might show up. Like they're really small, so if you stop,
Oh,
you might
take
just
a minute and we'll see.
potentially. Whoa, wait a minute. Is that a Piovius?
Medicine don't make you.
That is the little thing that just went up.
Oh.
like this spreadsheet though.
Huh. It was swirling around the arm,
I mean,
so it's probably swirling
I would say
around somewhere.
for
But it
individual, like,
looked very POBS-like.
because presumably there's some
We're at
trigger that she's
5:58. It's possible. Okay, so all I have to do is say, why haven't we said any of the polychaetes? And boom,
That's the magic
two of them
words,
just show up. Let's see some more.
Look at the thumbnails and you're like, all right, that one, that's enough of it. I'm not sure.
So the Pelagobia that we saw is annelid worm. Belongs to the family Lepidorhynchidae, and they are very sensitive to temperature changes when we have them in the lab. Oh, that might be that one. They're interesting because they're very short little worms with long parapodia to swim with.
How many R's in
They are neutrally buoyant, and when they swim, their head flops back and forth in an incredibly awkward way that makes it seem like sensing anything with the head while swimming just doesn't happen. So they have some potentially curious biology going on. There's another species or another genus within that family that are parasites on fish or predators on fish. It's a little bit of a unknown, but their first three parapodia are modified and have big hooks instead of bristles. Oh, look at that. Little platycellus. So the first three parapodia, or essentially appendages, help them attach on. They're incredibly fast swimmers, and they swim around, catch a little ride on a fish for a bit, feed on their skin and mucus, and then swim away. They're cute because they have these very beefy appendages in the front. And the difference in swimming speed and elegance is pretty pronounced, given the same body shapes, but different morphology. I like the observation in the chat about it looks like outer space. There's no up or down. It's exactly what the animals out here experience. It really is truly a three-dimensional habitat, which is one of the reasons probably that we get such strange body forms with animals out here. They have to be aware of a large volume of water around them because predators can come from any direction, up, down, all around. We don't usually worry about things coming up out of the floor to grab us, but these guys do, and there's no floor. Oh, I think there might be a Tomopterus up to the left. It was a long, straight, totally transparent thing. Yeah. Basic nightmare.
Just when I'm looking away.
Little larvation. I only saw it at the last second as it disappeared, so it may have been really small and really close to us. So, if it didn't show up immediately when we started going up, it probably jetted away. They can be very fast. Yeah, so the animals out here in the Midwater, they have a very different environment that they have to deal with and all the challenges that come along with that. We live in a three-dimensional world, but we're pretty much tied to a plane all the time, whereas the animals out here are not. Let's see another POBS.
It's something like this, but instead of it being
also haven't seen any nemerdians that I know of, like ribbon worms. Come in lots of pretty colors. They're usually much shorter than benthic or nearshore ribbon worms. They're nemertians. That's probably another one of this. No, what is that? It's got funny little paddles on the side.
Yeah, it's inside already, so
I thought it was a worm, but it's not necessarily. Like a tiny ctenophore with huge paddles.
oh, look at that.
Oh my god.
It's cute.
It is I would guess, a bioluminescent pteropod. Tiny, tiny. Yeah, they're gymnosomes. They don't have shells. They eat the ones with shells. The little. We have not. We've gotten some in the toes. But not this kind. Super tiny. Heather would love to suck this up, but it is really small, so.
Good to go. Got it.
Do you see it in the other? Oh, okay, cool.
Keep an eye on it. Follow it. Get
We need to check this guy for bioluminescence because there is one in this group, which may be Theliptodon, that is bioluminescent. And it has a little ring around it like this one did around one end that is a bioluminescent band, which is, I believe, the only pteropods that have bioluminescence.
Already got half suction on.
Yeah, he's not, he might swirl away, but he's not gonna race away. He's here. Sorry, pointer. Yeah. Do you want the laser? He's outside. He's behind the arm now.
Anybody want dark chocolate?
Yep.
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 The thermometers to photograph is that medium-sized one towards the front of the shelf.
Yes.
Great.
With a yellow label.
Great.
I don't remember the sample number, but yeah, he's the only one in there. Yellow label. I didn't mean the whole bar. Dave.
Yes, you did. What time is this coming out?
2:30.
2:30?
You up for a plankton tow after that?
Yeah, plankton toe, and then
Yeah.
it's going to do a tucker.
That's the plan.
Okay. And the tucker's to how deep?
1,200.
Oh, okay.
We're gonna try towing a little faster.
Yeah.
Careful setting up this.
Crunch, crunch.
Maybe a couple pods
It's like chocolate nibs in this dark chocolate.
I will be here once again. Are you collecting water on this thing? No. You done? Yeah, you could have collected from the depths. Yeah, but I mean, you could come in here and fill in those holes.
Yeah.
And then filter all my water. Clean the ocean 1 liter at a time.
How do we know?
2 liters at a time. I did the back of the envelope calculation. You should be done with 1 liter.
Those were pretty cool observations.
Got a little bit of good footage, yeah.
Yeah.
I know it's just something
They're
that
just so tiny. So you give us really nice cameras, now we can see those things, and then we expect you to collect them with this bread truck-sized vehicle, right? In my limited experience of moving it around, it adds one more axis of something moving to have to keep in your head. So if you can get it out far away from the vehicle so you have less push from the vehicle water, like the package of water that's kind of getting pushed around with the vehicle, then it's better. If you were to just hold it out there and leave it in one spot, it would be fine. And then you can orient it somewhere that's easier for you to see or easier to fly into or something. Yeah. I'm game to try anything. Why? Yeah, just don't. Yeah, don't do that. I know doesn't help.
This micro move. It's really cool.
Just like it.
Yeah.
That would work. I think that would work. Oh, what is that? Oh, it's probably another rhizarian.
This is not.
Yeah, we have enough of those. Quick, before Manu sees it.
Get it on the screen. Get it on the screen.
Get it off the screen. It was a radiolarian. We already have 4, so he's cut off.
Were they all in the same canister?
No.
He took up 4 canisters?
I don't know. It looks like they're in at least 3 different canisters.
You will not, is what I heard.
I said if you were wanting to get more, just stack them on the same.
You check it down.
You found the photo? Oh, no. I was looking at the
They definitely went in.
Oh, I know that. I don't know if it went up.
I don't think they did. But no, we didn't check. We could.
You wanna check?
Should, probably.
They could have
Yeah.
a category up here that said not a whale. Look at every another not a whale.
Done.
Not a whale.
Be careful, we might see a whale.
I know, you have to have the one that.
That would be mine.
Oh,
Is that them sitting right there?
You have to have a whale.
Just sitting on the bottom. Yeah. It's that. At least one of them. Cool. Good.
You don't have whales in fathom lakes.
Well, do you have swordfish? Because we saw a swordfish at like 500 meters the other day, which seems deep to me. Maybe. I mean, I don't know. Yeah, it flew behind in the tether camp. Just like, just moseying, just coming to look at us. It was crazy. It was super cool.
He had swordfish.
From the whale.
Like attack it, hit it.
Oh, that's.
He broke his nose.
Yeah, it's not tail.
2 days later, we found him in the water.
No way.
Of course.
I was surprised.
That's crazy.
It's good. It's still alive. They're still alive.
Calicopteran.
You think the penguin one is nice?
Yeah, it's like,
Oh, Jesus, you must have grown up in a rough neighborhood. Oh my God.
Big over here on the left.
Okay.
That's the problem.
Oh, yeah.
But big is a relative term, right?
Yeah, it's kind of funny because one of the jellies we collected the other day was big. I totally thought it was much bigger than this. It's a nice little siphonophore. I'm not sure which one it is. Maybe not a siphonophore person. I don't see any little tentacles in between the swimming bells, so it's probably, I don't know, Mars. That's a wild guess. So beautiful tentacles.
Done.
look at those jets.
Right. front of the camera, so this is supposed to go good.
So probably the really bright parts that are really reflective are the nematocysts, or concentrations in nematocysts, like nematocyst batteries. I think we can probably leave him. Very tempting to look at all these little tiny dots. Colobonema up there. Larvation. Need to find some more big larvations. Bigger.
About 10 more movies on the
What's this guy? Wouldn't be a terrible thing to find a pig butt. It's an odd little thing.
right in front of the camera, so I don't know if we can even do anything.
Yeah, I think it's pretty tiny little dot. Good enough. Little solmithes. No, not a solmithes. Had so many tentacles spread out that it looked like it. Probably something more like a chrysota. Oh, so cute. That is a Halicera conica, probably. Very pretty little jellies. Keognath indeed. Pretty orange one. We are almost to 600, 584. Should be a few more things down here. very peaceful doing the water dives in the middle of the night. So peaceful that,
Yeah.
to be careful. Too many nights without sleep.
Tight schedule, huh?
Yeah. You know, it's really your fault for letting us dive as much as we want. Doesn't happen often, so you know, it's kind of like, okay, let's go look.
Take advantage of it, right?
Absolutely. What's that? Almost looks like a sinker, but I think maybe it's just a ghost tail from a siphonophore. Or more big. Maybe there's a head to it. Oh. Huh. Looks like an apolemia.
Karen, could you pass me the coffee? What kind of things?
Absolutely.
Hold on 1 second, I'm not sure.
Oh, hey, Lex, what kind of produce did you get them? The ones with the long, thick arms,
No,
or the sparkly ones?
We got him one sparkly. It's in
Okay.
December. We got him two we haven't seen. One is a colony of two,
Wait, in
and one was a
December?
colony of
Yeah, you
five.
wanna do those in and then?
They're all in sections. So we didn't take any
So they're the
decent.
ones with the
They were
kind of
kind of
thicker
weird
ones.
sparkly that we hadn't seen, like
Okay.
sparkly, spiky,
okay.
that we hadn't seen before.
Oh, okay.
We did try going after one of the Coelodendron, whatever.
Okay.
I need to see it written down. It's not sticking.
I
in my
know,
head here.
I know. None of them are sticking to my head. He needs to give us a little intro talk to all
we
of them.
probably need to print a little cheat sheet out too,
Yeah.
because every once in a while
That would
I'm
be
trying to
super
remember
useful.
what's like.
Yeah.
It's not so bad when you're not kind of at the mic, and when you are at the mic and you're not sure,
Yeah.
it doesn't make us look
No.
good.
No, but we can't know everything.
And we can't have Dougal in the room all the time. So
I think it is an apolemia. I thought I saw a tentacle there, but maybe not. We can leave it. It's very pretty.
Okay, cool, because I'm the next one, I just didn't know when. 1:30. One, right, okay. See.
Oh, I haven't seen that before. My antenna doesn't look like it goes to the boat, but it's just, see it over there.
Yeah,
It's just really trailed out.
that's just our,
Ah,
trail.
that's your path. Okay. Oh, I see. Right.
Yeah, see it.
So obviously not your
Nice to get a
tether. That would be pretty cool.
visualization. Yeah.
You can see those little loops. Oh, not that. What is that? It's moving very fast for something mostly transparent. Maybe it's a fish. It is a fish. Never mind. Probably, cyclothony or luriglossus. Cyclothony. Based on the fins and the big lower jaw. Let's see who that is. Rosacea, probably. Yeah. We don't have to do that. We don't have to look at that guy.
Skip it.
Yeah, we've gotten a couple of those.
it's a prey of siphonophore.
Oh, look at that. It's probably too small. When things are a little out of focus, they get these strings around them that make you look like, oh, look, it's got wings. But not really. Oh, copepod. Maybe? No. It is.
Increase that.
It's a copepod. You know, if we had like 8K, it would just be jumping on you. It'd be popping.
I couldn't identify.
It's another acloptolid. That's just
That was cool. We can leave it. It's too small. Don't expect me to say that again.
Oh, the focal point separate is local.
Little Arctopodema or something like that.
It's not one of the major, let's see, class, order.
There's our tactical. Right, the backward.
Oh, right, I keep forgetting my mistake. Not starting it, not starting it. Classical reporter.
Hyperid. Cruising. They are super fast swimmers. They're not exactly the most aerodynamic animals. It's like a swimming bee. They do have a super cool trick. That was a platycellus, and they can roll up in a ball and lock their tail in place so that they don't actually physically have to hold themselves in a ball. It's a little defensive strategy. Makes them harder to chomp on. Of course, it does them no good if they just get sucked in, which is how a lot of the fish eat. So, but still, must be good for something because there's two different families, three different families that do it.
So that changed my version, so we can watch the ship.
Doesn't seem to be much current tonight. Is that true?
Yeah, it's pretty
Like,
good.
super straight.
Is pulling us
Yeah.
forward or depending on where it is. So I think if I shift my heading a little bit, it will pull us backwards instead of forward.
Oh, two different kinds of radiolarians. The same frame. Both of them giant. And a siphonophore. It's a radiolarian. No, don't call Manu. Oh, okay. Well, then we have a twin Manu in the D sampler. I'm surprised there isn't more hyperids on things around here, on the jellies and stuff that we're seeing. There also isn't that. Oh, that's a cute little jelly. What is that? We're definitely looking for a doliolid tonight. That might be a single one. It's a bit unusual. They're usually in a chain. Have to get a little bit of zoom, yeah.
Sir?
Does indeed look like one. As long as it's just not just a funny angle on a scythe. Nope, that's definitely a doliolid. What do we want to do with that? Shall we suck it up Yeah, we have 3 buckets left. It's an awfully valuable thing.
Yeah.
Yeah, what's easier, suction or deep? This guy's not gonna run away.
That's a good question. Then, would you recommend descent or suction?
Well, I would just.
Who's saying no?
Oh.
The desampler?
But you don't know what.
Desampler have a slightly bigger opening.
Yeah.
It's 5, right? I'm gonna try it.
Yeah, I don't know.
D2.
Alright. I got it right from the porch.
I'm gonna follow you down.
Roger.
It's a doliolid.
The camera a little bit.
Doliolida.
Roger.
Where's Russ?
Wait for you to get up a little bit higher.
Come up ahead, come forward. Come up ahead, come forward. Just stubborn. Nice. Yeah. Up. 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 It's in now.
It's in. Nice. It's in there. Right there.
Hey.
Woohoo! Nice.
Yeah.
Yep. It's in the can. Excellent. Right, so these are done. Unless we decide to drop the protist if something really spectacular comes along, but
Yeah, let that shrimp.
2 holding hands, nah. 2 of them. Twins. Do we have suction on?
No, but we can put it on.
We can we put it on and just see what we catch for bycatch?
Alright, we got it on half.
That also, oh, we don't have the shadow cam. Never mind. Won't flow things through the shadow cam. Maybe I have to ask for polychaetes again.
Yeah.
Saw those 2 right after I said it, but not another one.
Let me see.
Yeah. Oh, so cute. Its entire mouth parts and tentacles comes all the way out of the bell every time it pulses.
So tiny. Just to be able to angle it in.
Do you want it at like an angle that way so you can see both, or just so you can kind of push it a little bit when it?
Slow.
Oh,
Or we'll just have it.
what is that bouncing thing over there? It's a very odd behavior. What is it doing? Is that a jelly pulsing? Just carrying a bunch of stuff? Is it a Dumbo octopus? Japatella, maybe? It's definitely behaving a bit like a squid.
Stream yet. I don't even see it on the sit, then, do you?
No,
No.
It's not on 60.
It's that far away. Oh,
Yeah.
okay. Just used to things being way closer.
It is a squid.
3 havens.
It's like a cephalopod. It is a squid.
That is so cute. We need Dr. Judkins. That is quite the odd behavior.
Seeing that vision?
Maybe iris down a touch. What a weird posture. Looks like he has a photophore above his eye instead of below. It is
Sure.
very tiny. Maybe it's a baby octopus?
It's an octopus.
It's
Japatella or Volatina.
It's like so tiny.
Mm-hmm.
So
I'm
they
gonna
start
say
out
that
looking
tentatively.
like regular octopus, and then they get all. I
I'm
don't
saying
know how
this
you would describe
tentatively. It's
Japatella. I
got
would
no
say-
fins. The squid have fins.
Yeah, which is why he's bouncing.
Uh-huh.
I
He
thought he
is
had a photophore
adorable.
above, but it doesn't. It was just reflection. He is super
I
cute.
wouldn't be sad if we tried
Look
to
at that
collect him.
siphon.
He's probably gonna try to get away. If we could get some good video first.
Yeah, this is beautiful. Can we iris down just a touch? He does not like our lights. Look
Yeah,
at his
no.
look at his irises. I think that's
And the
his.
reason
I
I
think
think
that's his grumpy face.
Japatellas because those eyes are huge.
Yeah.
The Volatina pygmea is a sister,
Mhm.
and that guy has small eyeballs closer to
Okay.
his little head.
Okay.
Oh my god, he's adorable.
He's super cute.
It's one of the pelagic octopus species, insuri, which means he doesn't have those fins. I think so. I think that's the thing. Look at him.
So cute.
He's small though, huh?
He's very tiny. Yeah. We just saw a little bouncing
They get
ball in
the
the
size
distance.
of. Who cares about this Solmissus? Every time there's a jelly that comes in with a cephalopod. Cutie.
So cute.
Why don't we try this one? I
Ah.
want to see his. Come on. Mature adult.
Oh, it's inside.
Yeah, she's close. There, she's back out. Or he.
Yeah.
At least one.
I would say if we could.
Can you zoom out?
I don't know where he went.
I think that. No, that's not it. That's a shrimp.
Back.
That's not it, is it? No. That was great video.
Yeah, that's a great video. Is that him?
There's the Solmissus again. You know,
Yeah, but he
just
was
in
right
case.
on the other side. Great. He was still up to the
Sometimes.
left a little bit.
Great. Great to drive by.
A little guys.
He was cute.
That was great video.
I don't think so.
Fantastic video.
Really nice.
Thank you. Thank you. I guess if you have to pay attention to the Solmissus, it's fine.
That's okay, we can move on.
More saps. There's more saps.
More subs for sure.
More steps would be good.
Karen, I'm coming for you in 10 minutes. Is that true?
But, sounds good.
Okay. I'll finish up.
Can we
Thank you.
I love an octopus sighting at 1 a.m.
open the iris a little bit?
That is going to be in Fathomverse tomorrow. I will review.
It's a good one.
Before I go to bed.
Stole the computer. Are we going to be doing any more PIVs? Or are we just going to be saying?
No, probably not.
Haven?
I mean, if we
What's
saw
up?
something really, we see a large larvation, you know, we would do it, but otherwise, probably not.
Did you put the tooling cam on this one? Perfect, right? The tooling cam? Yeah.
think it's here. I think HD3.
Well, that was exciting. A little baby, probably Japatella. Pelagic octopus. They live their whole life up in the water column, drifting around. When they get a bit bigger, their bells get a lot more rounded and a lot more gelatinous. They have really, really soft bodies. So misses. Long time no see.
Yeah. Thank you, Evan. Perfecto. See if you can make it. Okay, coming forward.
You have to be certified first before you can start.
661 meters. Mycerian and a Calicopteran. We don't need either of those. As tempting as they are. Also haven't seen any isopods. That's a little odd. I know there's a few over here. Ooh.
Sort of geofenced areas, so specific coordinates, say Caribbean,
I've not seen that many squids, other than all the omostraphids up at the surface. Like none of the cranchids. There's a lot of really beautiful, totally transparent squid that we usually see. Cute little jelly.
Yeah.
Oh. Little scythe down below. Whoa. Good spot. I'm like looking over here. Oh, there's a thing right there. What is that? That might be a Pelagobia based on the shape, if it's fuzzy. It is a Pelagobia. Tiny orange worm, that would be great.
We're going to try a selection that's down here.
It's wee tiny.
Okay. See it on the sit. You got half suction on.
They can swim when they want to.
You want to catch it in the last moment?
What's that?
Last moment. He swims out. Karen said they swim.
Yeah, we
They're
can
pretty
do that.
steady swimmers though.
Okay.
Not that that's
I'm gonna
really
turn it
helpful,
off.
but
Yeah.
Get close.
I'm walking them in a little bit so you got the same plan as that. It's huge. What are you talking about?
really? Because I can't really tell it from all the other specks out there.
Yeah.
Oh, it's curling up a little bit. Oh, there's a free swimming Phronema down there.
I think we're maybe a little bit above the funnel now. Hit it. There you go.
Oh yeah.
Go in.
Didn't even have to extend it. It looked like it went in. I couldn't see through around the D-sampler,
Yeah,
but
just
definitely went.
stop.
Looked like it went in the funnel.
Okay.
Okay. Some decks in. We're on jar 7. Is that available?
Yep.
Good.
Good. First one of those. Usually we see a couple of those in the trawl, but they also don't like temperature changes. So if we got them up from 600 degrees, where it's only 7 degrees, 6.5 degrees through 29-degree water, they'd probably pop. That would explain why we haven't seen any of them. fairly consistent response to hot temperature.
They would pop.
Yeah, they just all of a sudden they go.
A lot of times when they feel the suction, they swim on.
Yeah.
So then turn on the last one.
Swimmer.
It is. For Nima. I don't think we need that one. I have a couple of them in the behavior chamber right now, possibly. Oh, what's that though? Do you love it how we pick out different dots out of the 1,000 dots on the screen? Oh, that dot, that's the one.
You guys are remarkable. I guess you have to recognize
Some of it is imagination.
some optimism.
Yeah, exactly. Very, very optimistic. I think it's a little safe. Can't quite tell. You what? Oh, I think that's another one of those worms. What is that?
Come on, autofocus.
Oh, I don't think autofocus will work, probably.
Maybe it's right.
I think it is right in the camera.
Alright, it's too close then. I'm gonna try the back on.
That is. What?
There you go.
I don't know what that is. I don't know what it is.
Mystery thing.
It's a mystery thing.
Roger.
It's a wee tiny thing.
Roger.
Might be a scythe, just a couple of bells, large and all tucked up together. Yeah, I think that's what it was. I think it was a teeny tiny scythe.
Yeah, that is
Oh.
in our camera there.
Yeah. When you can only see them when they're 3 inches in front of the camera, you know you're in trouble.
Yeah.
Sorry.
Yep. Alright. Continuing on.
That is
Pirouettes down there.
exactly. That is a funny swimming pattern.
I love shrimp.
Like to eat or to watch?
They're my, like one of my favorite things to see down here.
They are really cool, especially the ones with the really long antennas, right? That are. So somebody did an experiment with them one time, and they took, a piece of like fish muscle or whatever, and coated it in green dye,
Yeah.
and then they dropped it. And they had a bunch of Surgestids, which are the shrimp with the really long antenna. They had a couple of those in the tank, and they were swimming around. And as soon as they hit the trail, you could see the trail of where the food particle fell because of the dye.
Yeah.
They'd be swimming along, they'd hit the trail, and immediately follow the trail. It was really cool. That's this guy.
What?
Sorry. That's why we keep them.
This is what you missed.
Guy still.
That's why I
Oh,
said you had to be
it was
there.
head off of a fish. Tricked us. Tricked us. We have not seen a lot of fish.
Oh my.
I mean, other than cyclothony.
That is sick. I know. I can just throw an SOI logo on that.
What's your first thought?
Well, because I understand.
Excellent, excellent job.
Thank you. That is amazing.
Oh.
That's swimming.
Oh, yeah, that one.
Cost way more than my reputation.
Oh. It's like controlled falling. I think it's a hyperid. Slightly drunken. Obviously had his units for the day.
Yeah.
bizarre swimming behavior. Some of them do when they are disturbed or light happens, they'll roll up in a ball and their response is just to drop. They're really negatively buoyant
Oh man,
and they just fall really fast.
turn into a pebble, huh?
I mean, we see that in a very small tank, but it's interesting to see it in a larger context. Sure, it's platycellus, or as some others say, platyschelus. Where'd he go? Oh, he's inside camera.
Sinking.
Super fast sinking, right? It's a
Yeah.
bit like a little stone. There he is.
There's two of them.
Oh, double points if you get
Is
both.
that three? There's three on frame.
Okay, triple points. Yeah, maybe we just kind of stop and hang here and see if they come to the lights
Okay.
and crank the suction.
Suction on jar 7. Coming up to full.
You guys all come fast.
How hard do you think
I think it's better
full
off
is?
shutting that off front if you want to join ventures. Get the nozzle close to the full suction.
I'm hoping on for maybe some bycatch of them. They're not super abundant. Oh, look at that jelly. We've gotten a couple, but there was a lot more around when we got some before as bycatch.
We're open for very delicate things. I'll crank the suction down a bit more.
Okay, yeah. Yeah, these guys are like little tanks. They should be okay in high suction, even if they hit the screen.
You want to try to get one of these?
That would be great.
Okay.
They're pretty wily and pretty tiny.
I'll leave it camera, okay?
We just gotta pick which one we're going for, I guess.
Oh, twofer. Twofer. These guys are cool. They have 2 pairs of eyes. There's one. What's that? No, not remotely. And they fall like stones
Yeah.
when they fold in.
I have to crank the
Yeah.
suction. You got it?
Thank you.
Would it help to put the nozzle out a little bit or no?
Oh, no.
Oh,
That's better.
oh. Bang shot. There's more. There's one. Oh, he's right in the front. Come on, guys. It's fun in here.
Hang out.
Really. We'll show you movies.
But I know like 3 hours of them just behaving. Something with the sea level, it's pulling in a lot of air. Close enough to the pump. It would dry up and
It's raining them now.
It is.
That's the thing, it was working. Up until the pump taking out the end of the channel.
It's
So then it
above
was just like,
and behind us now.
We are at 6:10.
There was a slight leak, and then I
Get it back to the middle.
under the glass, and then I see it on the side of the glass or something. Also the LED.
Is there any way to measure that sinking speed? Like you don't know how fast the vehicle is going up and down or anything, would you?
Going down 28 meters.
Just let me know where everything is.
Down at 2.8 meters per minute. That's pretty fast.
Lined up. Lined up still. We're looking good. We're looking good. We're looking good. Looking good. Just passed
Come on,
it.
buddy.
Passed it.
Oh, he's just inside. I thought he was exactly dead ahead.
I thought we were looking at the word dead ahead. Is that what you were looking at, Carrie?
No, we're trying to collect this platycellus. But this one could just swim right in if he wants. That would be okay. No, he's really close. He's inside. What is that? Is that a squid?
Yeah, it is. Ditch these hyperids. Forget the collection, my friend. You might hang around. You might be okay.
Yeah,
You might be
might. Do you think it's one of the Omestrephids? Is he
I'm working
inside?
on
No,
that.
he's outside. Oh man, he's just sitting on the nozzle. That's just rude.
It's in the DPI.
He's just teasing you. He just wants us to go look at the squid. There's one.
Man overboard.
There's another one.
Keep pointing. Keep pointing.
Just leave the suction on. They're bound to get a couple of them. There's another one.
Are you as unimpressed by a lot of these? You guys are too small. Vision language, architectures in Mayan.
I'm still hung up on anything else. Yeah. I don't know. I'm not so. I'm behind the times.
Did you hear that? They're sinking at 2.8 meters per minute.
No, I mean,
Sinking at 2.8 meters per minute.
Solving the main
Super fast.
Do you know?
Because that was the sub was going down to keep up with it.
We have many other problems.
but in terms of like
Like
flexibility
9
of
feet
missions
per
that you
minute.
can apply to the vehicle.
Just doesn't want to do it. No, he's not. He's not swimming. He's just falling, but I think he's maneuvering because you see his tail come. Oh, now he's swimming.
Right, like yonder.
Do you think? Let's hope. Maybe you can see our lights reflected in his back there. There are several of them around. Whoa! Whoop! So we're not going to index. We'll just let this. We'll maybe put a couple more or something else in there. I don't think he'll find his way all the way out.
There he is.
Woohoo! Oh, he's big.
Yeah.
Alright, so leave it
Straight to the screen.
Which is also a strength.
Grab another one.
Yeah, let's grab another one if we can.
Fill the whole jar.
Maybe I farm out. Maybe I farm out.
Doesn't that-
They have super cool eyes. They're, supposed to have one pair of eyes, but they have two. But they're not particularly specialized eyes that we can tell, that we haven't mapped them just yet.
I'll give you two.
It's gonna like with the gonna be over-engineered in a certain ways, but just for we don't really make stuff for production level.
We do.
Oh, just inside. And he's off. He says, I'm out of here. I don't like the feel of that.
It's getting the data out.
No, but there's not even the hardware that can survive for.
But if it just goes through the whole of the boat, I mean, if that's all
That's
his,
trickier.
if all his, no, I know.
That's okay.
But what if we did?
I think most of these big dots are probably them.
I think something like that that goes through the whole of the boat.
I'm here to relieve you, but you seem very excited about your hyperids, so we can, I understand your mission.
I was assuming that squid is the same one we see at the bottom.
No, it's not.
Since we're at
It's
600
not, it's
meters
different.
instead of
Yeah, that was a different.
200
Maybe
meters.
I just tell him.
Nope, yeah, he was definitely different. I think he was an awful too,
Get
but
some water.
I would love to.
Get some plumbing, do a flow through system and just.
They're probably long gone anyways.
Probably.
That would be pretty impactful.
Yeah, sorry, we were late.
No, it's fine. No, we should stay with the hyperids. You guys
Laser
need them.
focus.
Makes sense. Happy to stick with them for a bit.
What
Need them.
everybody has.
The goal is to what? Hang around here until buckets are full, aren't we
Suck
supposed
up
to?
several.
Right, so we can pawn it
I don't
off
think
to-
we hunted on the way down, did we? I think they
Paul
mostly
coming out
Came
kind
down.
of came down
And so
fast
slowly
and
up.
yeah. Well, we still haven't
Oh.
reached.
What is our depth?
Oh, 620. We have reached 600. Okay,
Great.
we're good. Things just got interesting
I
though.
know, right?
I would
Hanging
just
out
keep
down
working
here.
and then we can bomb up
Yeah.
to the top.
You want
And
to
it's with a,
You can leave it on at like 30 or something. Keep them in there. All right, you're here to relieve me.
If
So we
you want
have
to. Yeah.
2
Yep.
buckets.
Okay.
These are, but if we need to drop something for D, it would be this.
Okay.
Although it's
Do you want to
holding
drop
hands
this?
too very nice.
Oh, that's the one he
I think that's a
Yep.
generous
Yeah,
option.
exactly. It was one of these.
Yep, it's the dots. It's the hold hand dots.
Yeah.
The whole hand dots.
The holding hand
They,
dots.
they were. They really were. It was like that,
That's my
right?
kind of.
Yeah, there's something coming up.
It's like
They're super cute.
Falkor.
So
That might-
that's
Okay.
what's in there. I wouldn't
And is
drop
there any
it,
other
but if
priority
there's, like,
besides the hyperids?
worms,
No, I mean, it's
larvation. Well,
massive
we put stuff in these already, so no larvations.
Okay. So worms, fine.
Far right.
Okay.
Um,
And we
yeah.
got his whole
Swimming
hands,
thing.
so we're good there.
Really good opportunities to inform
we had a,
how to make.
you know, whatever you see that looks.
Dougal's happy for the moment with his.
He's got
Yeah.
plenty of
I was
stuff.
gonna say he's got plenty.
But yeah,
got
we
a
see something weird.
whole-
Be a flow through system.
Whole bunch of jars he needs to take care
Yeah,
of.
he's working on
Um, yeah, so whatever
Okay.
looks interesting.
Yeah.
Flow through
I can send you the list of stuff we've already collected as far as jellies, so that you can
Yeah, but
like
that's
those
what
and
That would be helpful.
plumbing
Yeah, that'd be
system
great.
parts you can get off the shelf to replace things.
Yeah, thank you.
we can set up our own.
Thank you.
parallel
Of course.
Ben?
And then
You're most
ending,
welcome.
our ending time is 2:30?
2:30.
Okay, on the surface, 2:30.
Yeah.
Okay,
I'm starting to think more and more. It's probably.
easy enough. Let's take a look at this fish if he's gonna stick around.
It's just
Oh,
really nice
he's a
that
beautiful
so much
cyclophony.
hand-holding.
This is the most abundant fish in the oceans, and that's what they do. They swim away all the time. Yeah. And we already checked this guy out, or no? If we could, that'd be great. Oh,
They're so fast.
it's we've got this guy already. If we could just get a zoom on it. This is one of Dougal's. Do you guys remember the name of this one? It's the rocket ship one. Calicofrin. Siphonophore. Yep, thank you. Yeah, just get this for a couple seconds. Perfect. Great. Okay, we can move on. Thank you.
We could.
Calicofrin, Siphonophore.
I mean, all.
Should be. There's a whole Siphonophore section up here. Definitely not that. No. Up.
How would the data?
Yeah, perfect.
or
Yeah.
something
That I don't know.
They would just push it to either text.
Okay.
It still exists.
Yeah.
If we need a direction, we could head up a little bit. We're not in any rush.
Cool.
Yeah. Like copepod, I believe. Yep. If we can zoom, just grab them for a sec. I think it's a copepod. I don't think it's a pteropod.
Oh, sorry. I'm gonna try to back up a bit. then.
I did, but I didn't see him super focused, so I didn't catch him.
He was really tiny. We
That's what I
didn't
thought.
quite look at him, but it looked like the bioluminescent.
Copepods. Yeah, that's
Had, like, a
cool.
little ring around him.
Oh, cool.
Yeah.
Yeah, and we collected him or tried to?
We tried.
Okay.
He was too tiny.
Yeah, that's what I was thinking. Okay. The search continues. Yep, perfect. Looks syncerish to me. Yeah, we're good. Let's find that little squid that was hanging out.
Yeah. An opportune time for it to appear, right?
It's what they do. It's what they do. This guy. Another fish. Ketigna? Yeah, we take a look at both. This is a fish. It's a different fish. If we could, that'd be great.
fish or on that other one behind?
The fish, please.
What was I doing?
I actually like things with a backbone. No, I'm just kidding. Just kidding.
Grabs.
He's really small.
Yeah, he's small.
What's the?
We're good. We're good. Let's go down to that
Hey, Dan,
siphonophore.
what is these?
Well, you took an observation of the fish.
I don't know. I did have to zoom in a little more. I missed it.
Yeah, this guy down middle low. There's quite a bunch of stuff here.
Yeah, when they start walking, then it might be decent size.
Yeah, you want to see his. I have his slides. He sent them to me.
That's it.
Okay. So it's one of the Bisonex siphonophores, right? Look at me.
Yep, that is.
Look at me getting in.
It looks Nanomia-like.
Yep.
Paging Dougal.
Yep. He's working somewhere. I saw him a little bit ago. Yep. Get a look at that. The red feeding down here, and then the top of the bell, whichever's easiest to grab first. This is great.
And it's not Yeah, this looks like a Nanomia.
Fabulous.
We've been missing them. Maybe we're reaching.
These sort of sequences.
Shouldn't be too close. It's not even on the thing.
It's pretty.
He's great. Any chance for a little more zoom on the head portion up at the top?
Yeah. Good.
Thanks. Yep.
Wow.
Great. Down low. Yep. Yep. We're good, you guys. Thanks.
And there you go.
He's like, I've had enough.
Okay, we can keep going.
Yep.
These
We
are
sure
great
can.
shots.
This is good. Perfect. Yep. Exactly what we wanted. All right. Let's move it on. A little
Missed it.
fishy.
Did you see that one?
No, I'm troubleshooting the shutter cam. I think my USB battery must be shelf life while we were here.
No.
Just doubly making sure that's what the problem is. Having to swap it out of that.
Little house. We're
I
good.
have a question. Do you have a good?
From multiple
Sinker. We're good.
process on there about more coming up.
Do you want to recruit some help?
USB helping us use
Were we up here?
signals.
This guy? Is that what you're looking at?
Or maybe almost, yeah.
Oh, sorry.
No, no, you're pointing at the right thing. I was trying to.
Between the two. Yeah, yeah.
They won't be the same species.
Oh, yeah. What do you mean? Which white dot
I
is
don't know.
which
We
white?
just came across one that
Little
reminded
dot.
us of an anomia.
Right.
It didn't have tentacles in between the ectophores.
Yeah, because we had said we were going to collect more hyperids if we could, correct?
Yeah.
In 7? Yeah, yeah, yeah. Perfect. Yep. Yep, yep. Absolutely.
Love how you've been
So
following.
bitter still.
But I don't know if that's something you would want to collect or anything. Because we've now come across, I think, 2 or 3 of them.
I could probably do calls today.
All right.
Unless there's something we really already know we did, and I don't have to, because identifying a bisonex is quite a time consuming versus other cases.
Okay.
Is this a...
Is it? Look, we just asked you. We identified the move.
Our walking reference. Okay.
So Dougal, the one we were looking at is the other option. His eyes aren't right. His eyes are too small. And the octopus one.
Okay.
Rock serious. I agree with you. I don't know. But it's not. Here's pteroctopus. I want to look at the video again. That's Teropus. Those eyes were not bad.
Images of a live pteropus.
Better. And it's not
And the
scarred.
eyes were a lot bigger than that.
These guys are close. Right. These
What-
are our other two options.
What's our max depth tonight? What do we want?
Around now, 600 was.
Oh, okay.
Mhm.
Do we want to go
Uh-huh.
slow?
Karen said hang out here for a bit and then slowly kind of work our way up.
Okay. There's a Tomopteros or a worm thingy. This one?
Where do you have them?
This.
She's got center middle. Yep.
Sorry, Hillel.
No, it's perfect. What do you got? You think this one?
No, I
Right.
have no idea.
Right. I need the video. This guy, his eyes are too small.
Yeah.
I don't think it's
Did I
him.
get on the informer?
What is it doing?
Yeah, but do they? Maybe.
It's like a dog chasing its
We're gonna
tail.
we're gonna continue this quest.
Can
I will
we
keep that
zoom-
on as the ID with a
Can we
question
get
mark.
a really good zoom on that?
Yeah.
We will do our best.
As you always do, my friends.
Yeah.
That is not.
Must be a Tomopteros. It doesn't have the squiggle tentacles.
Alright, let me block it in, Ben.
Feel like a
Yes.
lapel.
Mm-mm.
Still out a little ways. Do you want to hit lowers, Ben? Lower so we get some more light on it. And if you kill uppers and mids, maybe.
Better. Better. There he is.
That's better. Is that a
Yep.
baby Tomopteros?
Definitely Tomopteros.
Whoa.
Yeah, he had some bioluminescence out of there. That's great. If we can collect him, that would be awesome.
Suction it.
Yes, please.
Not in jar 7. That's where the other one is.
Are the Tomopteros, was there already a Tomopteros in 7?
I think one of the little things.
The hyperid amphipods are in there.
Yeah. I see it in the SIP, but I can't. Yeah, maybe pop those other lights on real quick, Ben.
Oh, there's another hyperid.
Well, I had to look at the new specimen pictures. It is.
Is that him here?
Similar to something Rob just sent me.
That's too far.
Medium elevation.
Something he got from the bed.
See him?
That he's,
No.
Can't figure out what it is except the keys. So it's the same thing. Only this specimen's a bit more.
Okay. We can keep the quest. We got some video real quick, so we got that.
We didn't sample that one, right?
We did not. We can't find him. He has evaded us. All right, so let's keep scooching. We can start moving up slowly. Would be great. Up, forward, slow.
Oh, right, turns around right when there's a siphonophore in front of it.
Was there? I must have missed that.
Huh?
I must have missed that.
Darn. Oh, darn. Straight ahead, that looks like something Manu would be interested in.
Yep. Here.
For me yet, Di?
Looks like
Yeah, I think there's a larvation in there.
Where?
If I'm not,
Is that
where are
true?
you looking at?
I
I
think it's zipping out.
didn't somebody. That's a copepod. I think somebody put one said it was in a. Is that not true? Oh, it's a doliolid in the decoupler.
Well, that's still something
Still a
for
tunicate.
Okay, then yes, we have something for you.
Oh, gosh, I thought it was going to go to different end.
Just kidding. Don't look too disappointed.
Now, that, on the other hand, is probably a fritillaria.
This could be a
Zoom in.
fritillaria.
It's a Doliops.
You think?
Belongs
Let's take a look. Let's take a look.
to waiting. Belongs to this area.
Yeah, you might have to go lowers again on this one.
Yes.
I like doliolid.
Doliopsid.
Yeah,
Oh, it is a
doliopsis, right?
Doliopsid.
Sid. Thank you.
Yeah, he's right in the camera.
Well, it's all
There
right.
it is.
Didn't see bioluminescence.
We were just conducting an experiment.
Impromptu.
There.
Mm-hmm.
Belongs to ricerium, this.
Hmm?
Belongs to ricerium, no.
There you go. Tunicata.
Ah, tunic.
That's great. And bye-bye. Are you happy with that, Russ?
Yeah.
Sweet. And there's a crustacean. I think we're okay. Russ, you got enough on that, yeah?
Hmm?
You got enough footage on that?
Yeah, good enough to tell me it's a dolly hopsit.
Okay.
Otherwise, not much more I can do without a specimen.
Got you. All right. We're moving on, my friends.
Swimmer.
Who's that?
It's an amphipod.
Yeah?
It
This
looks
guy?
Like a
Stomatopod?
Foramina.
Amphipod?
I suspect it's an amphipod, like a
Yeah.
pteranid.
If you could get back to that little guy who just went off to the left.
Oh, here's the Tomater's egg cage.
He might have swum right by us.
This is one of my favorite.
Yep. Yep. He was close.
Around the camera.
Yep. No worries. Okay. Let's go back on track then. We'll go slow forward up, but if we catch that siphonophore on the way, that'd be great. If not, we will keep moving.
Where's David? This is the
You want to stay on jar 7?
Yeah,
And-
that's the hyperids, right?
Can
Yeah.
we do this?
If we see any other hyperids, of course, we can stop on that.
We're on deck at 2:30.
Yeah. Rumor has it. We have an hour. What?
Recovery from 500.
We don't have any Niskins on this one, right? No
No
Niskins?
Niskins to check.
Okay.
I don't know.
We're
I
working our
feel
way
like
up.
it's pretty amazing.
So if we were to just recover from 500 meters, that would take about 40 minutes. Just a point of reference.
Yep.
Without stopping.
Do you guys want to slowly get up to 500 and then get to the surface, or would you like to go shallower than that? The deep scattering is at about 300 and above at this point. We don't spend much time. Is that what you're just saying? Yeah, that's what I was saying too. Okay. If we could go up to 300, let's try 300.
Risaria.
If we could go up to 300, keep your eyes closed for a minute, everyone. Or 5.
Straight up to 300.
Wait a
Way
second.
belly
What
to
is
the
that?
left. See what it is?
Yep, left corner, is that what you're
Right on
saying?
the edge.
Yep, we got you.
This thing is coming into the
Yeah,
center.
this little guy.
Little bison neck?
I don't know. He's little.
Or is it a collapsed toast?
I don't know. That was very solid.
Hit it, Dan.
That's weird. It wasn't what I expected.
What is
at all.
this?
Okay,
Sinking, yeah, I was gonna say
Go ahead and leave it.
sinking house kind of thing. Okay.
Yeah.
We're going up forward to 300. Quicker than slow. If that makes sense.
No, I'm here to slow
Sorry,
it
you
down
guys.
even more. I'll go get Dougal.
Yes, that'll
We can, yeah.
take us about 12 minutes if we hammer it all the way up.
To 300.
Yeah, that's just going straight up.
Then that's good.
Any samples left?
I think so.
Sit out. That's all the way. There we go. Thank you. And if we just get a slight forward on it, it helps with the snow. We're gonna go up to 300. We're not gonna stop, and then we'll hang there for a little bit. So Dougal, or Russ, you have 10 minutes to not call anything out, except for an observation.
Do I have to go get duct tape, put it over my mustache?
No, because that might be
Because
painful.
that really hurt. Get it back off.
That would be painful. Don't do that, my friend.
find a totally opposite colony or stop right
Nope. I might even say no to a squid. That's where we're at. I'm here for the team.
I doubt that.
I'm here for the team.
Why? Why do you have to
Multimedia
do
will
that?
always lead the shots to the squid.
Why do you have to do that?
Because I don't want to be the biased one just because I happen to be sitting here.
Karen already did that.
Both
Don't worry
of us,
about
yeah,
it.
both of us. Alex is hunger.
We could always
For the
pull some
cephalopods?
B-roll if we
Yeah.
need to put some
Nice.
squid in.
Oh.
Oh, I just got bumped to the B-roll? Wow.
They're saying that we're not fun.
I don't know how I feel about all this.
Thanks.
There we go. That's the kind of excitement I like to see over on Twitch. Submarine time.
That's how we feel in here too.
Yep, that was totally opposite.
And that was squid ink.
It begins.
Here we go. 1 for 1 so far.
Gonna tally up who misses how many things?
It's fine. It's fine.
So right now we are just booking it up to 300 meters or so, coming from just passing 550 right now. We've got about more than an hour left before we need to be back on deck, so making our way up into the scattering layer and see what's up there.
Deep scattering layer. They should be up there.
Yeah. See a little surface.
At 300, there might be bigger ones.
Yeah, I just keep thinking of the little squid party we get every night.
I know.
Which-
Those little guys are adorable.
Nothing wrong.
And those are juveniles.
Nothing
Those are not
wrong with
adults.
them, but they're little.
Oh, they're little. I agree. They're really cute.
That one that self-sampled itself was like the size of my pinky.
Very opportunistic. Yeah, no, he was like this.
Had cute eyebrows though.
He was great.
We could be collecting dinner.
Yeah.
But we are not doing
We'd
science.
be out there jigging for squid right now.
Could.
International waters.
Mm-hmm. It is pretty fun. I've caught Humboldts that way. That was fun.
How come they're so dumb? There's no hooks on those.
On those squid jigs?
Yeah.
Yeah, there are.
That's right.
Just
Those things are
pins.
deadly pins.
Yeah.
Yeah, they're fun.
Oh, and sub 5.
Oh, really? Like suction sampled?
No, we just I just go underneath it, picked it up, and close the lid.
Oh, wow.
100.
That's cool. Yeah,
You saying they taste good? Is that you?
they do. They're delicious.
Yeah?
It's all right.
The thing with animals is you have to catch them within the first 3 minutes because they're blind, can't see them.
Oh, when you got a sub.
No, near. Even that's how I catch most of the in the first 5 minutes, 10 minutes. Fine.
Just done it like that.
Yep. Yeah, I think that section on the
Just because they're under talking. Are we there yet?
Almost. Look, kids. Big Ben, Parliament. Some rain, snow. We're getting there.
Cyclothony or Ketic
I think it was a cyclothony. That was what my first instincts on. Looked like it had fins. Unlike the Ketic gnats.
Is the ceiling leaking?
Who's angry?
Is there water on my leg?
Yeah, they're big. The ones we caught were like 4.5 feet, 4.5, 5 feet in total length. Yeah, it was great. And we caught a bunch all at once. I went like 19 nights, 0 squid.
Yeah.
Like Heather doesn't know how to fish. She can't catch her own research. She can't do what she's supposed to do. And then the last night, in like the last 20 minutes, we caught like 12 of them or something. It was great.
Saved.
Yeah, it did. It saved me. It saved the research project. It wasn't my research project. I was just collecting. So
That must have been pretty frustrating 19 days.
we had only 1 hour a day. 1 hour a day while the CTD was going down. So
Water just
Small
appeared.
it was a small window. There were 3 of us, I think, jigging each night. It was fun.
caught a lot of octopus.
Yeah, I haven't caught those on jig yet.
What's the Laxophobia?
Thalassophobia, that's a fear of the deep sea. Well, fear of the ocean. Deep water. Do you like swimming in just open ocean?
Maybe. Who knows?
You probably don't have
Who
it.
knows?
Last one was cute. I did it once.
That little guy down low, I think he was different.
Just look at all the stuff we was
Deep bottoms water.
Terrible, Ben. Terrible.
They do quite a bit in filming too.
No rules then, man. They were very rigorous.
Ooh, that is the first, was that burrowing or was that?
Oh,
it was the green-tail guy, too.
heartbreak in the rooms.
It doesn't matter.
I'm gonna get
Whatever it
I'm gonna
was.
take it off this. I'm gonna get removed if I have
Yeah,
too many of
we're gonna
these.
talk about all the
Doesn't matter.
the world-changing animals that you pass by.
None of it matters.
They
None of
are.
it matters.
Because
All
they
of
weren't-
them are.
All
Nobody
because they
cares.
they had less than 8 legs.
Nobody
No, no. Good call on the arms. And no. Being very impartial to
Arms,
all of
legs,
us right
same
now.
On 400.
Sweet. Just a few more minutes to go. We'll stop at 300 and explore around there for a bit.
And why is it we're stopping at 300?
At 300 right around now is the edge of the deep scattering layer, the bottom edge, and that is an aggregate of organisms that come up to the surface every night under the cover of darkness to feed Some migrate vertically from just a couple hundred meters, and some will come all the way up from 1,000 meters or more every night up into this epipelagic zone, which is 0 to 200 meters. So we're just gonna go just below that to see who is there. Getting there. We're getting there.
Almost there. Recovery.
Almost there.
Yeah, I can't decide. Hmm?
Medusa. We're not. I'm
Not
just
stopping.
pointing it out for the observation.
No.
Just pointing it out.
Meters.
Count them down. Count them down from 50 items.
All right, chat. Count down those meters. We're getting to 300.
47.
348.
That won't be fun.
That's right. Don't mess up though.
I mean, the
Get them
cool
in.
thing
Get them in the
is
right order.
Going from the scan to the animal just disappearing.
Rhizarian.
But I mean, like,
Same Rhizarian, maybe?
Yeah.
Super
it's
close.
Spinning around and
I say it's
it
close
got
to the
cycled in
camera.
the hood.
Yep,
Yeah.
yep.
Oh, there we go. We got someone doing the countdown. That's-
oh, I guess
I
could
think
do an
they-
effect where you just do a fade to black and then overlay that so that it disappears when it.
Station.
Yeah, I think this one's really nice.
Yeah, Heather, you got 15 minutes.
Yeah. Shopper spree.
Uh-huh. You can do this.
That's
We can
right.
do
Just
it.
run down the aisles.
Yep.
Scooping all the
We're gonna
squid into your
scoop
cart.
everything we can find. We have 2 buckets left. What can we scoop?
That's right.
15 minutes.
Just huck it in the bucket.
Bucket in a bucket.
Sweet. All right. We are approaching 300. We're going to explore here for about 15 minutes or so before we've got to make our ascent to the surface. So you guys can open your eyes again.
Very nice on the countdown. I appreciate the excitement at 300 too.
Alright, forward and
Up
Slightly
slightly.
up.
Yeah, forward and slightly up would be fine. This is great. Maybe a smidge slow. Yeah. Yep. Fabulous.
Screensaver from 1998.
Yep, a gentle falling snow. That was a copepod. Top middle.
Yeah.
Looking to see if this is anybody home. Yeah, somebody's in there. There's something in there, but I don't know if it's the actual animal or something else. This could have been a house situation. If you want to focus in on that siphonophore as well, we've got the house to the left and a siphonophore over to the right. Yep, here's the siphonophore coming in. I don't, is he close? Or is he far?
There we go.
There he is. If we can get some good video on this. Julietta, you can get this, yeah?
Just a bit better than. I was asking you what your.
A beautiful one. Yeah? Yay. We're getting there. We're getting there, Lakshmi. We're getting there. Yeah, this is great.
It's hard to pick, right? The lowest gets the backscan, of course, stuff.
Okay, we'll just sit here for another couple seconds, getting some images here. If you could zoom, and then we're good. Let's see. Not gonna agree with us. There he goes. He is keeping that whole movement going.
Just
Getting
testing new eyeballs, man.
It's all good. We've got some video of him. We're good. Oh, that's one of her hyperids, though. This guy.
That guy?
Yep. What are the chances we could collect him and we could keep him in the same bucket 7 if that is an option? This little dot way out here. He's up off my screen. He's back on. This little guy. There's a few of them actually, I think, but let's stay with him. Yep. I've got him still too. He's out of focus for me. Better. Thanks, you guys.
So now here's the question.
He's zooming. Oh, he's staying.
Science camera footage from SOI. Our laser. What levels do we
He's here. Coming down. He's just going down, down, down right now. Yep, he did. We just hang here for a minute. They might come back over. If we could. Is this guy? There he is. There's one.
Oh, there's a bunch of them.
There's a guy.
Definitely that.
I lost him again back in here.
It's more
If you could put the science cam, so I have a little funnel in the corner. If I see anybody, yeah, like that's great. If I see anybody close over there, let's give it a minute. There's a bunch around here. That's good. This guy, that's one. I don't know what the chances are. He's all up on this camera.
Let me know if he's close and I'll zoom in.
Okay. I think. This is one, or maybe it's copepod. Can you zoom on this guy? Yep, still on my thumb.
It's close to the funnel and then zoom closer.
Yep, that'd be great just to see. I think it is, but I'm not 100%. It might be a copepod. He's small.
Not even picking it up on a stick.
Yeah, nope. You're good. It was a copepod. Okay, we can move forward and slow again. If we could put the cameras back forward. Thank you. Perfecto. And then slowly up. Thanks. Sinker. You could go forward a little more if you wanted to. Hey. Middle?
Those three things.
Guy? I think there's a siphonophore below him, but let's stay with the guy in the middle top.
There. Zoom in this here.
Are both the D samplers full?
Yeah. Is this a Medusa or is this a house? Looks like a house. Yeah?
What's this behind it?
What's this?
Big
That?
siphonophore. Yeah.
Is that big siphonophore
Yeah.
behind it?
Yep. We can go to the siphonophore, get some video. Do you want the house? Russ, I don't know what to do. They're
Well, you're
literally
not
on
gonna
top
be
of
able
each
to- You're
other.
not gonna be able to collect that house out
Okay.
with anything other than
Let's
D
get some
sampler, so-
video and zoom on the siphonophore. It's huge.
I think Yost is asleep, but if he wants to DPIB or anything with this guy.
Almost big.
Roger, push the one easy.
And then if we could zoom when you get a second, that'd be great. Yeah.
So let's, are you okay with us, taking a look at this thing?
Sure. The
I'd like
only
to
thing we have to
start-
consider is we're going to come up with an empty bucket then, because we only have 5 minutes left before we have to ascend.
Okay, then don't worry about it.
You sure? Just get some video on this end, and then the tentacle end. If you could zoom in a little bit on the head end here, nectophores.
There's another really long.
That's
siphonophore
great.
here.
And then if you, I don't know what the chances are, the ability to pan up him. I figured.
In case you wonder.
This is great, you guys. This is amazing.
It is a siphonophore.
Nicely done.
One of the most amazing animals in the ocean.
Gorgeous.
Defisenect siphonophore.
Fisenect,
Look
right?
at
I can get Fisenect.
Irena.
that.
E-R-E-N-N-A.
There he
That
is.
the genus.
It's a fish eater.
Just
This
let him
guy
go
is
out of the frame.
gorgeous.
That was an amazing
Lovely
clip, by the way.
pass. Nice job, pilots.
highlight.
And the siphonophore agreed with us.
Big space. You gotta press the
Get
green
your
for
last little
resume
tentacle
feed.
out of there.
Yep.
No, you-
Nicely
Thank you, Tyler.
done. Nicely done, you guys. We've got 4 minutes, my friend, so we're gonna move
Oh, there you go.
up and slow.
That's what you want there.
Well, it's the
Unless you
same
want this
siphonophore, isn't it?
Oh, is it?
Yeah,
Well,
isn't it?
I don't know.
Feel free to take a look,
we haven't
but
got
We have
a
one
sample from
sample.
here.
we only have one container left.
He's gotten so far
Is
away.
it suction or D?
It's suction. Okay, let's get on this guy. Isn't he the same? Are you sure he's not the same animal? I think it's the same animal.
Got real far away real
Could be the
quick.
same
He's
species.
pretty large, my friends. He's shedding his little, what are those? Rock?
Gorgeous.
Yeah.
tentacles.
I'm trying to figure that out.
Tentacles?
Okay, I need,
Yep.
need to get it
Look
against
at
the
how
vehicle.
Oh yeah, the bracts. Yeah, shedding the bracts.
Yeah.
Yeah.
Yeah, it's the same animal, my
It's
That
friend.
called a
blackish pigment
nectosome,
is
which is an
distinctive
assemblage
of
of
the
nectophores.
Dougal gets the closing images of this beautiful siphonophore.
Check it out, twisting.
Yeah, he's gorgeous. It's gorgeous.
Wow, look at that. It's twisting itself.
Wow.
To change direction.
Eats fish, Dougal?
Yeah.
Yes.
Only?
As far as we know, yeah.
Yeah.
And surfers.
They have a close up of the tentilla later on when
Thank
you
you.
get a chance.
What'd you say, Dougal?
The
It's
tips of those
losing.
tentacles.
You want to zoom in on the
Bracts, is
tips
there?
if you
Yeah.
can? If you guys can zoom on
See
the
if they
tips of
have
the
a little
tentacles.
red spot. Like
Back on.
So there's a lot of the individuals have.
Here you go, Dougal. Does that help?
Yep, that helps.
I don't see red tips.
I don't either.
Roger. Or you can leave it tuned if you want. I'll just bring it in.
These guys probably would hurt if you got stung by one as well.
You think so? I would think.
No, just
Yeah,
let me watch.
so let's
I
get it
don't
all,
believe
let's get it all.
One way to find out.
Wow.
Do you want to zoom in, Dougal, in the middle?
Yes.
Yeah, sure.
Keep going.
Yeah, just keep doing that, yeah.
Go and suck water from around it without sucking the
And we will get some bracts and stuff, yeah.
You get enough material.
That's
Look
great.
at
Still have lights up
that.
Yeah.
Stuff like
Yep, if there's
Well,
stuff
if we're
lying
gonna do
around, we can get in a
that-
jar 7, that's
Yeah,
fine.
suck up
We-
some of the pieces, I
Could we
guess.
do
Yeah,
a quick
do you want-
scan then?
Yes,
Into
All right.
jar 7's okay, yeah?
Can we do a scan then? Because I didn't think we were going to collect any material, but
Okay.
if we're going to collect material around the animal, I would like to get a scan.
Okay. We're gonna scan with DPIV.
Yes, please.
Not gonna fit in iris.
No. DPIV.
So we're gonna DPIV this quick, and we're gonna collect some of this stuff around. Whichever you think is easier. If it's easier to suction first or DPIV, you guys choose. I don't know how long he's gonna shed this stuff, and I know we don't have a lot of time.
Yeah, we are at a cutoff time, so
Yep.
do whatever you guys deem priority. We'll do that first.
Do we need DPIV? We're at cutoff time. So
All right,
tell me what
go-
you need, my
Go
friend.
suck.
Suck parts over DPIV.
Yeah.
Yep. Suction parts. That's the answer. That's what we're doing.
We want to get right up close. I've got light section on. We want heavier just to start grabbing stuff, or what?
Dougal, what do you think here?
Want to see the long things floating up on the left-hand side? There's a-
Like all of these things can get sucked
Yeah,
up.
there are bracts.
These are parts of it that it sheds. So if we can suck those parts up, you don't want to touch the animal, right?
No.
Yeah, exactly. So let's try to get this shed.
Oh, stop.
Hey, we're asking people to collect single cells. You guys can collect some shedding.
Still say, stay on the same jar?
Yeah, same jar. 7. We can stay on the same jar with the hyperids.
Yep.
We could just
Yep.
turn it on.
And then I would just turn the suction on as we're getting around it.
The tentacles take out.
The tentacles would be nice to ID the
We
species, but I
get
think I
the
can do it by
side cam down
as
the
well.
bottom. Put the board down here.
Yeah, if we can get any of all of that. Oh, there's some going in. Or did that go behind?
Okay.
I'll watch here.
Look at the paper and look through and see what they're actually.
And then just keep me honest, I'm not bumping into the
Yep.
creature's sail.
The sequence of the
Oh,
bubbles.
there's one that just got in.
So just leave that little lean
Yeah.
alone.
Yeah, if we could, that'd be great. Guy behind you, gently.
It's so big.
Here's another
It's
big
so
one
big.
coming.
We're not going to increase it,
It's
just.
so amazing.
Yep, yep. He's still shedding,
So
so
amazing.
there's a whole bunch.
It's an earthling, you guys.
Is that just?
Yep, yep.
I missed part of
We
it.
don't think
Is it
it's
just shedding because it's shedding, or
No, we
whack it?
No, we
dropped some dynamite.
No, we didn't.
We did not
It seemed like
touch
a good
this
idea
animal.
because it was midnight. We'd all been out for a long time.
We turned the speaker up on the heavy metal and it just went, oh, not this time of night. That's, you know what time it is? It's 2 a.m.
Okay, thanks for that
Look at
answer.
that.
That helped.
I don't think we've
Don't
sucked
think-
anything up yet.
We got one
We
piece so
can-
far I saw go in.
Did we hit it? Or did
No,
it-
the animal?
Oh, it's
No.
over there. It's out past the line.
No, yeah, animal's
Right,
fine.
but those are,
We got
don't think
about
those are
30
masses
seconds
or
left, my
anything
friends,
on-
for attempts because we're at the cutoff.
We could definitely get that.
That is a good piece though.
That's fine.
yeah, I don't see it.
That part, that is the end.
But if-
That's the end. We're not getting
From what?
these. Not these. Maybe.
Yeah, I feel like it's easier to target.
They're just with the currents, yeah. They're just floating around.
Things.
All right, my friends, I'm not sure how successful this is going to be.
Pump on.
It is. Suction's on, right?
Can we try and go after one of those worm-like pieces?
That's what we were just trying. The current keeps scooching them. If we could get one of those, we could call this.
That is gorgeous.
Yeah, is
I
nearby.
feel so privileged to have seen that.
Now we're just getting picky. Yeah. I think if we want the pilots to pilot for us again, we have to go to the surface.
Hopefully we got something, right?
We might have gotten a piece or two doable, but I'm not sure. Oh, do you have to? Okay. We might have a piece or two. All right. Thank you guys. So if we could head up to the surface, that'd be great. Let's put this baby on the ship.
All right, so that is going to be us moving to recover. Thank you for joining us for another dive stream. This is not the end of the cruise, though, so we're hoping to get a couple more dives in. Keep your eyes peeled. And I'll keep the cameras running while we make our way to the surface, and hopefully we'll see you on the next dive.